Good morning, everyone. Welcome. يا مرحبا. MashaAllah, I can see the number of participants is increasing. So I'll give them 3 minutes to join more, and then inshallah we can start. Welcome, everyone. Good morning. نورا الحين نبدأ. All right. Bismillah ar-Rahman ar-Rahim. With respect for those people who came on time, I will start, inshallah, this session. As you know, guys, we have 3 days sessions that is focusing on microorganisms or microbiology and related with infection control, infectious diseases, and how we get a better understanding for the microorganisms, because I believe that most of you guys are not microbiologists. Yeah, since or from the fact that most of infection control people are coming from different backgrounds. So in this course, we focused on microbiology, or let's say clinical microbiology and general microbiology, and to understand the microbes, their mechanisms, how they invade a host. And I believe that if you get a better understanding for the microorganisms, you will get a better ways, or you will figure better ways to prevent the infections. From the concept that most of you guys are from nursing, public health, maybe other specialties, maybe doctors, not microbiologists. So understanding the basics of microbiology, very pure basics, could lead to better interventions. Yeah, I believe. So good morning, everyone. I'm glad to be again with you after our last, if you are the same people who attended last time. Last time we have done a very nice course about healthcare-associated infections for 3 days as well. And this course is related to microbiology and immunity. So by today, we are going to talk about microbiology in general, me and Miss Sumaya as well. So I'll have the morning shift, she will have the afternoon shift. As you knew me, if you knew me well or new to me, I like to have questions within the talk. I like to keep it interactive, kind of educational method. So you will not be bored, and feel free to join at any time. Just write to me your question in the question and answer session so I can reach you quickly. And at the end of the session, of course, we will have a 5-minute discussion time. And between each lecture and lecture, we will have 15 minutes a break for you and me, guys. All right. So basically, and very basically, we will start about resident and transient microflora. And what is microflora or microbiota in human bodies or in a host in general? From this concept, you can understand where does the microorganism come from, how they initiated, how they transfer, etc. So the object today is medical microbiology definitions, some of the essential definitions that it's good if you know them and get closer with that. Medical microbiology background briefly, I believe, and as long as we go further in lectures, we will go and talk deeper and deeper. And next, what is the resident and transient microflora in healthcare settings? How does that happen in healthcare settings? And scientific significance of the normal flora, how normal flora is significant to a human body, to a host, in a healthcare setting, how does that affecting everything? Normal flora of the body by parts. Of course, we will talk about parts of the body and what type of normal microbiota are inhibited in there. Microbial pathogenesis, how does these microbiota could cause a pathogenesis way in invading a host, or they are an advantage or disadvantage. We can see through the lecture. And next, sources of microorganisms. Like it's kind of knowing the story from where microorganisms are started. And I believe this is the most interested part. I have been studied during my bachelor, microbiology bachelor degree. Endogenous versus exogenous infections. So we need to know by the end of this session, what are the difference between endogenous and exogenous infections and the proper way, of course, to prevent them and the better way of understanding them. First, is my voice clear? Sorry, I haven't checked with you guys. Oh my God, yeah, thank you, Abdullah. Clear, thank you guys. Okay, good. So medical microbiology definitions, general concepts, okay? We are speaking from general concepts to a deeper ones. Okay, so general concept of significance of the normal flora being a normal flora, or what is the benefit of normal flora, or what is the effectiveness of their presence in the nature. So the normal flora influences the anatomy, actually influences the anatomy, physiology, and susceptibility to pathogens. Perhaps it can also contribute to morbidity of the host. So if you understand this concept, this minor foundation, you can understand how important is the normal flora in our life. So it influences the anatomy of the body, the physiology of the body susceptibility to pathogens invasion, and the morbidity of the host, and how the host can tolerate an infection, or to be a carrier, to be infected, or to be colonized. Yeah, so from there you can understand the basic core component of microorganisms is the normal flora in the body. So what is the skin flora? They are varied environment of the skin results in locally dense spare populations with gram-positive organisms. So mostly in skin flora, normal flora is gram-positive organisms, such as معروفة, we all know it, Staphylococci, Micrococci, diphtheroid. And usually they harbor on the skin, and they cause no harm to the skin because they are the normal flora to the skin. But whenever you see a gram-negative bacteria on the skin, and this skin has a wound or the barrier has been defected in the skin, this gram-negative bacteria could cause an infection or could invade the host. So gram-positive organism is fine, is a normal skin flora on the skin. If they are present with a certain concentration, if the host immunity responses is balanced, and many factors are there, making the host stable, then this organism will be fine to be present on the skin. And actually, it is necessary to have some bacteria on the skin because they protect the skin from getting infection from other organisms. So next is the oral and upper respiratory tract flora. As you know, oral and upper respiratory tract and gastrointestinal tract and the neuro tract are open tracts, and they usually received plenty of microorganisms from the environment, and they have to have a strong microbiota or normal flora. طبعا النورمال flora هي نفسها, it's the same of microbiota, but recently they call it microbiota in research and scientists, but before they call it normal flora, بس هي نفسها, same concept. And I will talk English and Arabic at the same time whenever I remember the Arabic part, okay? So because I think we have non-Arab participants, so I have to keep talking in English. And whenever you feel you're not understand, just let me know, but write in the question and answer so I can get you guys. Oral and upper respiratory tract flora. We need to have multi-type of flora in this tract so we can balance the non-wanted microorganisms, if I could say, non-wanted. So we can protect the tract from being invaded by pathogens that are harmful to the tract. So a varied microbial flora is found in the oral cavity, and the streptococcal anaerobes inhibit the gingival creeps. The pharynx can be a point of entry to the microorganisms and initial colonization for Neisseria, Bordetella, or Corynebacterium and Streptococcus species. So those bacteria are mostly the normal flora for the respiratory tract and the upper, let's say, upper respiratory tract. Those bacteria. Okay. Gastrointestinal tract flora, and logically, if we are going to talk about gastrointestinal tract, we have to determine that this place or this location of the body is a huge place containing a huge number of bacteria, and it got the highest number of micro burden in there. So we need to have a strong microflora or microbiota in the gastrointestinal tract to protect this area from the invasive pathogens that might be present. Organisms in the stomach are usually transient. Why transient? Because they came from the food, they came from the water, they came from outside source of the body. So they are usually transient. They are not usually there. I mean, they entered the body within the food, the water, the things that we eat and drink. So usually the stomach has a transient microorganisms, and their population are kept low as much as possible based on the acidity of the stomach. So the acidity level of the stomach is the first barrier in the stomach to keep the level of microorganisms in the stomach under control. Otherwise, the amount of microorganisms will be high, the organisms will be multiplying, replicating, and causing probably inflammations or infections or ulcers or etc. So the acidity activity in the stomach is the first layer of defense in the stomach and can keep microorganism in a low concentrations or controlled concentrations, we can say. Helicobacter pylori is potential stomach pathogen. So the Helicobacter is a pathogen that is potential to the stomach that apparently plays a role in formation of certain ulcer types when it's not controlled. The large intestinal contains moderately mixed flora. So the large intestinal, of course, contain the most or the biggest number or the majority of the microorganisms that could harbor in our body will be in the large intestinal. The flora of the large bowel is dense and composed permanently of anaerobes. They are لا هوائية, bacteria لا هوائية عادة, anaerobes bacteria in the large intestinal, because the oxygen is minimum in that location, of course. These organisms participating in bile acid conversion and in vitamin K and ammonia production in the large bowel. So this kind of flora is even impacting on the vitamin levels, ammonia levels in the large bowel location. They can also cause intestinal abscess and peritonitis, those organisms. Urogenital flora. The vaginal flora changes with the age of the individual. For example, the vaginal pH level, for example, and hormone level could be the balancing factors in that area, detecting the presence and absence of the flora of that area. Transient organisms in the vagina will be the Candida species, frequently causes vaginitis. The distal urethra contain a spare mixed flora. The urethra contain mixed flora usually, and these organisms are present in urine specimen, could be found, unless a clean catch midstream specimen is obtained. Conjunctival flora. The conjunctival area harbors a few number of microorganisms, or usually no organisms will be there. Haemophilus and Staphylococcus are among the genera most often detected as an infections in this area. So now when we can say the host is infection, and this is not anymore a normal flora, not anymore a microbiota that is living with the host cells in a balanced way, in a helpful way. Many elements of the normal flora may act as opportunistic pathogens, meaning sometimes the normal flora become an opportunistic pathogens, especially in hosts rendered susceptible. Susceptible hosts, those are patients with a heart disease, for example, or immunosuppression disease, or patients under radiation therapy, or patients under chemotherapy. And those people are very sensitive for normal flora shifting from being a normal flora to an opportunistic pathogen, because the host characteristics has been changed. The host has become weakened, and whenever the host become weakened, the microbiota or normal flora can invade the host itself, and they call in this case opportunistic pathogens. Of course, the body, Abdullah, produce flora, and by the end of the session, you will see the answer, maybe in the last 3 slides. The flora of gingival surface cause dental sometimes caries in about 80% of the population. So sometimes the invasive procedures in dental, for example, in dental care, can cause or reflect the microflora to be a disease or an opportunistic pathogen instead. If we are going to discuss about medical microbiology background, more to understand, so I can say Drive's microbial flora is associated with the skin and mucous membrane of every human being from shortly after birth until death. And this is maybe the answer for you, Abdullah, it could be. This bacterial population consists of normal microbial flora. The normal microbial flora is relatively stable with a specific genera populating various body regions during particular period in the individual life, meaning our after birth or during the human birth, the microbiome level, amount, burden on his body will be, or her body will be changed by time. Like the organisms, the microbiota during birth is different than during adulthood, is different than during being very old or elderly people. These changes of the microbiome level on the body could affect the human or the host by interaction with the microorganisms. Microorganisms of the normal flora may aid the host by competing for microenvironments more effectively than such pathogens as, for example, Salmonella species, or by producing nutrients to the host. Maybe they can also harm the host by causing dental caries, abscess, or other infectious diseases, or may exist as commensal by inhibiting the host for long periods without causing detectable harm or benefits. This is the relationship between microorganism and the host, and how the microorganism could act within living with the host. It could be combating the microbes environment around the host and protect the host. It could be harm to the host by causing disease, abscess, infectious disease, for example. Or it could be exist as a commensal without any benefit or harmness causing to the host. So these are the characters that microorganisms can be sharing that character with the host while living with it. Even though most elements of the normal microbial flora inhibiting the human skin, they inhibit in the human skin, nails, eyes, oropharynx, for example, gastrointestinal tract, are harmless in healthy individuals. So whenever the person is healthy, whenever the individual is having a balanced lifestyle, for let's say, these microorganisms could help and enhance the personal life instead of being pathogens or invading the host. These organisms frequently cause disease in compromised hosts, meaning the microbiota could cause harm for the compromised host, for the weak host, more than the healthy individuals. Viruses, parasites are not considered members of a normal microbial. So normal microbial, they could be most of the time bacteria, not viruses or parasites. Flora, by most investigators, because they are not commensals and do not aid the host. Whenever viruses or parasites are there, they are invading. Be sure that they are invading the host. They are never give the host any kind of benefits because they are obligate. They are obligate microorganisms, and they cannot live with the host in a peaceful way if the host is healthy enough. Okay. The normal flora in humans usually developed in an orderly sequence. After birth, leading to the stable population of bacteria that make up the normal adult flora. So from birth to adulthood, the population of the bacteria as a microbiome or microbiota is different in the human life. The main factor determining the composition of the normal flora in a body regions is the nature of the local environment, meaning there are some factors that impact on the volume of the microflora on the host as a carrier. For example, the local environment of that host, which is determined by pH level, temperature, redox potential, and oxygen availability. For example, water or dryness. Is the environment watery, humid, or dry? For example, this will affect and impact as well, and the nutrient level as well. So based on these factors, the volume of microorganisms of the microflora will be present or absent on the human, based on the availability of these factors. Other factors such as peristaltitis, the mucous membrane, saliva, lysozyme secretions, and secretions of the immunoglobulins also plays a role in flora control. So body secretions, body liquids, mucous membrane also can modify and create a certain environment, certain let's say volume of the normal flora on the human body. The local environment is like a concrete in which one principal instrument usually dominates. For example, an infant begins to contact organisms as it moves through the birth canal. This is the story of the microflora, how it's started. A gram-positive population by bifidobacteria, lactobacilli, for example, in the gastrointestinal tract early in life, if the infant is breastfeeded. So if the infant is breastfeeded from the mother, he will get another type of microorganisms which be inherited in his body, and could be shaping the microbiome level in the baby's body. This bacterial population is reduced and displaced somewhat by gram-negative flora. Let's say Enterobacter bacteria when the baby is a bottle-feed. So bottle-feeded will get more negative flora, gram-negative bacteria flora than the breastfeeded child or baby. The type of liquid diet provided to the infant and the principle of instrument they used of this flora control, immunoglobulin, and perhaps other element in the breast milk may also be important in shaping this flora. Then is the significance of normal flora. Some human studies suggested that the flora influences the human anatomy, as I said, psychology, and lifespan, ultimately cause of death as well. So the microflora could shape how the body is tolerating diseases, how the body is handling infections, inflammations, strength and weakness of the body parts could be determined by the type of microbiota present on that body. After getting the understanding or overview of the resident and transient microorganisms, now we should ask what is resident and transient microflora in healthcare settings. For example, as a healthcare worker, this is what we care about, but we need to have a better understanding. That's why I started with those slides in the beginning. First, hands contaminated with transient bacteria pose a significant risk for transmission of infection. So the problem with the transient bacteria more than the resident bacteria, because as we said, microflora or microbiota is a resident bacteria. If the individual is healthy, if the environment is balanced, if the immune response is strong and effective in a human body, the resident bacteria will cause no harm to that human being. However, transient bacteria, what we care about in healthcare settings, is the most important thing that we need to focus on and we need to prevent whenever it occurs. Hands with dermatitis, for example, or other type of skin breakdown are more susceptible to become colonized with transient bacteria, because transient bacteria could be moved by hand washing, by hand hygiene, hand rubbing, for example, hand scrubbing as well. However, when the skin is breakdown or the staff or healthcare worker has a dermatitis, the broken skin is colonizing the transient bacteria in there, and it cannot be moved within washing or hand hygiene practices. So this is the problem, which, including even multi-drug resistance, could be present in that skin breakdown. So preventing dermatitis and practicing proper hand hygiene whenever possible is a key role, or is a key factor actually to prevent cross-contamination and infection among healthcare settings. Some facts about resident and transient microflora in healthcare settings. For example, as we usually talk about hand hygiene is the most effective single practice that we could use to prevent infections. If we took a hand as a part of the body that could be a transfer the microorganisms from person to person. So skin hand flora, in the case of resident flora, these are some facts about it, and transient flora, some facts about it. Resident flora survive on the skin more than 24 hours because they are resident. This is their home. Okay, they can survive more than 24 hours. If no effective hand washing, hand cleaning, they could be survive on the hands as long as they can, because this is their nature house. Okay, nature host, nature home, nature place, if I could say. Not easy to be removed, even hours of scrubbing. Okay, complete sterilization is impossible from these germs, but they are low virulence. They have low virulence type of pathogenicity. Examples of staphylococci, diphtheroid, mostly they are gram positive, as I said, on the skin earlier. However, very few of gram negative bacteria. Whereas the transient flora, they survive on the skin less than 24 hours because they need susceptible environment to multiply, to survive, etc. So they survive less than 24 hours, luckily, and fortunately, easily removed with soap and water or alcohol, any practice of hand hygiene. Acquired during contact with contaminated areas like mouth, nose, perineal areas, genital areas, catheters, urinals, patient care equipment, etc. They may have high virulence on a human body. They can invade, they can cause infection, they can cause colonization, etc. Examples, Enterobacter, gram-negative bacilli, and Pseudomonas. How does that started? The whole story. Back into the significance of the normal flora. Although the foregoing indicates that bacterial flora may be undesirable, usually as infection preventionists, we undesire likely to like bacteria present. However, studies with antibiotic-treated animals suggested that the flora protects individuals from pathogens sometimes. So sometimes the presence of bacteria in the body is desirable because they can protect the body from some pathogens when they invade the host. Investigators have used streptomycin to reduce the normal flora and have then infected the animals with streptomycin-resistant salmonella. Normally, about 10 to 6 organisms are needed to establish gastrointestinal infection. After that, antibiotic course. However, but in streptomycin-treated animals whose flora is altered after the antibiotic course, fewer than 10 organisms were needed to cause infectious disease. Further studies suggested that fermentation products, acetic and pyruvic acid produced by the normal flora inhibited salmonella growth in the gastrointestinal tract. So some acetic butyric acids that produced forming from the presence of bacteria in the stomach, they can kill salmonella, some of the salmonella species, and protect the human being from gastrointestinal infections. If we could see this picture, we can understand how can this figure shows some of the factors that are important to the competition between the normal flora and bacterial pathogens. We could, from this picture, understand how could a pathogen invade a host. Imagining this is your host and this is the mucus cell, okay? This is your normal flora. The white circles is your normal flora, whereas the black circles is the pathogen. Okay, so if you have a strong mucous membrane, a strong normal or thick normal flora layer, for example, on the skin, in the stomach, whenever that surface is, if the pathogen trying to invade these layers, they couldn't, they have to go through many layers to could cause an entrance or a portal of entry to the host. But if you imagine if there is a less amount of normal flora on the surface or none even, so the surface is exposed immediately to the pathogen to invade the host, and it's easily can cause a disease. As you can see, the more black dots you have, the more disease likely to be happened to the host, whereas here the less, because they cannot fight all these kind of layers. So this is a kind of a protection of the disease and pathogens invasion. Normal flora of each body parts. So we need to understand each body part normal flora. From there we can understand how the normal flora could fight infections within that part. As an infection preventionist, we can get a better view, we can get a better understanding of what kind of bacteria that we need to enhance in our body or focus to have, or we are fine to sow some bacteria from the specimen lab, specimen cultures. We know that this is not infection, this is normal flora to be present in this sample from this body site, if you understand what I mean. Because sometimes we get, for example, a urine specimen, we have Candida albicans as a result. Okay, this is in the urine is not an infection indication. It's not an infection indication because it might be a normal flora. As long as the patient has no sign and symptoms of infection, then this could be a specimen that we are not looking at or not considering. This is what I mean. If you get a better understanding of normal flora on the body parts, then you can decide which specimen that you should focus on, what kind of result of this specimen that is really need an action from an infection preventionist, or it's just a normal thing to have. So first is a normal, normal flora, skin, I guess. Yeah, skin provides a good example of various microenvironments. Skin regions have been compared to geographic region of Earth. The desert of the Forium, the cool woods and the scalp, the tropical forest of the armpit. For example, the environment that the person is living on could determine what type of skin flora could be present on the person's skin. The composition of the dermal microflora varies from site to site according to the character of the microenvironment. Of course, different bacterial flora characterize each three regions of the skin. There are three types of skin that we need, areas that we need to focus on, and they have different types of flora. Yes, all of them are skin, but they have different type of flora. First, the axilla, premium, and the toe webs. This is number one. Number two, the head and face and neck. Number three, the upper arms and legs. Skin sites with partial occlusion, axilla, perineum, and the toe webs harbor more microorganisms than do the other areas have. These areas, number one, has more microorganisms, microbiota, than the other areas of the skin. These quantitative differences may relate to increased amount of moisture or higher body temperature and greater concentration of skin surface lipids. So based on the moisture, the temperature, and the lipids amount on the surface of the skin, those organisms are disseminated on the skin. The axilla and the two webs are more frequently colonized by gram-negative bacilli than the other areas of the skin. Okay, the number of bacteria on individual skin remains relatively constant, unless bacteria survival and the extent of colonization probably depend partly on the exposure of the skin to particular environments and partly on the innate and species-specific bacterial activity in the skin. So the number of the bacteria relatively is constant on the skin. However, whenever there is an intervention on the skin, for example, poor hygiene, personal hygiene, extra hygiene, this will disrupt the constant amount of the microbiome level or volume on the body. Flora on the skin. Staphylococcus usually is a major inhabitant of the skin. And in some areas, it makes up more than 90% of the resident aerobic flora on the skin. So on the skin, we have Staphylococcus epidermidis and Staphylococcus aureus as well. Staphylococcus aureus, the nose and perineum are the most common sites of them. They colonize with the Staphylococcus aureus, which is present in 10% more than 40% of normal adults, meaning that Staphylococcus aureus is the highest colonization skin flora you could see in a host. Staphylococcus aureus is prevalent 67% on vulvar skin. Its occurrence in the nasal passages usually varies with age, being greater in the newborn, for example, less in adults. So, and Staphylococcus aureus is extremely common, we can say from 80 to 100% on the skin of a patient with certain dermatologic diseases, such as atopic dermatitis, but the reason for this finding is unclear yet. However, it's a high percent on those patients with derma issues. Micrococci, also in the skin. Micrococci are not as common as a staphylococci and diphtheroid. However, they are frequently present on a normal skin. Micrococcus, the predominant species usually account for 20 to 80% of the micrococci isolated from the skin. Diphtheroids, usually, or croniforms, the term diphtheroids denotes a wide range of bacteria belonging to the genus of Cronobacteria, and extremely common in the axilla. It could be found extremely in the axilla, whereas non-lipophilic strain are found more commonly on non-haired skin. Also, streptococci is one of the normal flora of the skin, especially beta hemolytic streptococci, are rarely seen on a normal skin. The basicity of beta hemolytic streptococci on the skin is attributed, at least in a part of present of lipids on the skin. It depends on the lipids presence on the skin to be available, as these lipids are lethal to the streptococci presence. Other groups of streptococci, such as alpha hemolytic streptococci, exist primarily in the mouth, from where they may, in rare instance, spread to the skin. Also, gram-negative bacilli in the skin. Gram-negative can make up a small proportion of the skin flora, just a small amount of the skin flora, not just like gram-negative positive bacteria. In view of other extraordinary numbers in the gut and in the natural environment, there's scarcely of the skin is streaking. They are seen in most intertrigonous areas, such as toe whips and axillas, as we said before, and not on a dry skin ever. Okay, they need a moisture environment and a certain heat or temperature, heated temperature level, so they can replicate and survive. Dissemination of is the major factor preventing the multiplication of microorganisms, gram-negative bacteria on intact skin. Enterobacter, for example, Klebsiella, and Escherichia coli or E. coli, and pertussis species are the predominant gram-negative bacteria found on the skin. So those are the most gram-negative bacteria found on the skin. Acinetobacter species also occur on the skin of normal individuals, and like other gram-negative bacteria, is more common in moist areas founded. From the skin, moving to the nail flora, the microbiology of normal nail is generally similar to that of the skin, meaning the organisms that prefer to live on the skin, they prefer to live on the nails as well. But, however, dust particles and other extraneous materials may get trapped under the nails, depending on the nail contacts. In addition to the resident skin flora, these dust particles may carry fungi and bacilli. And from here, we can always re-mention and re-announce that having a short nails during your healthcare services is essential, because this will increase your hand hygiene practice, will increase the safe practices during your service to the patient. Aspergillus, Penicillium, and Cladiosporium are mucor and the major types of fungi found under the nails. So we don't want to give the patient Aspergillus, Penicillium, etc. Nails should be short during your service. Make sure this will impact negatively on the hand hygiene practice. So the nails could harbor not only bacteria, but also fungi. Next is the oral and upper respiratory tract flora. The oral flora is involved in dental caries and periodontal diseases, which affect about 80% of population in Western world, according to the studies. The oral flora, its interactions with the host and its response to environmental factors is quite important, because the oral microflora is transient bacteria, so that they came during drinking, during eating, etc. So they can impact negatively on the host. Anaerobes in the oral flora are responsible for many of the brain, face, and lung infections that are frequently manifest by abscess formation. Therefore, Infections carried from dental side is very, very huge and important because they can impact on brain, face, lungs. They can cause abscess formation in these critical areas. The pharynx and trachea contain primarily those bacterial genera found in the normal oral cavity. For example, alpha and beta hemolytic streptococci. However, anaerobes, staphylococci, Neisseria, diphtheroids, and other are also present and isolated in the pharynx. Potentially pathogenic organisms such as Haemophilus, Mycoplasmas, and pneumococci may also be found in the pharynx. So all these microorganisms could be seen and found in the pharynx and the oral upper respiratory tract location. The upper respiratory tract is so often the site of initial colonization by pathogens. Type of pathogens could be colonized in the upper respiratory tract is Neisseria meningitis and diphtherides, Bordetella pertussis, and many others, and could be considered the first region of attack for such an organisms, because whenever the host immunity is altered or weakened, those pathogens are going to be an opportunistic pathogens, and they can invade the host immediately whenever the host is weakened. So these kind of like upper respiratory tract harbor a very huge, important actually bacteria, which can cause several invasive diseases. In contrast, the lower respiratory tract, small bronchi and the small bronchi is usually sterile because the lower tract has many, has in between the lower and upper tract lymph nodes and immune response, high immune response. To maintain the lower respiratory tract sterile, clean as much as possible of the bacteria which can reach in that area, because particles the size of bacteria don't readily reach it yet, and if bacteria do reach these regions, they encounter the host defense mechanisms. As I said, the immune response is very high in the lower respiratory tract, such as alveolar macrophages that are not present in the pharynx and in the upper respiratory tract. Gastrointestinal tract flora. The stomach is relatively hostile environment for bacteria. Of course, as we said, for eating and drinking, etc. It contains bacteria swallowed within the food, and those dislodged from the mouth as well. Acidity lowers the bacterial count in the stomach, which is highest after meals, usually, and lowest after digestion process. Some Helicobacter species can colonize the stomach and are associated with the type of B gastritis and peptic ulcer disease too. Most of the bacteria cultured are streptococci, lactobacilli, bacteroid, are though to be transient as well. It is colonized with streptococci, lactobacilli, bacteroids, and bifidobacteria permanently. Gastrointestinal tract flora also, in this highly anaerobic region, let's say in the large bowel side of the intestinal, this genera replicate and produce metabolic waste products such as acetic and butyric, and those lactic acids could also maintain the balance of having healthy pathogens inside the large bowel side or large intestinal side of the body, because as we said, there is a huge number of bacteria in this area, and the more we have, positive bacteria, positive attitude, not gram-positive, positive attitude bacteria, they release citric acid, bitric acid. Those acids could minimize the replication of a harmful bacteria that's coming from food and drinks, etc., which are transient bacteria. The strict anaerobic conditions, physical exclusion, and bacterial waste products are factors that inhibit the growth of other bacteria in the large bowl. Other bacteria that we don't want them to be grow, they are inhibited by these factors. Although the normal flora can inhibit pathogens, many of its members can produce disease in humans if the immune response of the host is altered. Anaerobes in the intestinal tract are the primary agents of intra-abdominal abscess and inflammations. Also, gastrointestinal tract flora, anaerobe can also cause problems within the gastrointestinal lumen. Treatment with antibiotics mostly will impact on the gastrointestinal tract flora. That's why if the patient have long-term antibiotic therapy, patient will develop Clostridium difficile by time because the gastrointestinal tract flora has been disrupted and their positive function will be no more longer with the host, serving the host anymore. So treatment with antibiotics may allow certain anaerobic species to become predominant and cause a disease, unfortunately. For example, Clostridium difficile, which can remain viable in a patient undergoing antimicrobial therapy for a long time and may produce pseudomembranous colonitis. Other intestinal pathogenic conditions or surgery also can cause bacterial overgrowth in the upper small intestinal. For example, SSI infections, surgical post-surgery infections could also lead, could interact with the gastrointestinal tract flora and therefore cause infections in that site. In this situation, the patient usually has been compromised in some way. Therefore, the infection caused by the normal intestinal flora itself, and it is a secondary to another problem. If you could understand what I mean, the normal flora in the body will cause an infection, and this infection considered as a secondary infection to the main problem that happened. But first, at the first place, there is an intervention happen. There is an invasive procedure happen to the host which lead the microorganism to be, or the microbiota to be a pathogenic instead of normal flora. Urogenital flora, the type of bacterial flora found in the vagina depend on the age, pH, and hormonal level of the host. Lactobacillus species, for example, predominant in female infant because of the vaginal pH level approximately is 5 during the first month of life, or during birth. The pH level is 5. So the lactobacilli species will be the dominant place, predominant microorganism in the female infants. During adulthood, the pH level is changed from 5 to 7. The glycosin secretion is started. Therefore, diphtheroids will be started. Staphylococcus dermatitis, streptococcus, and E. coli also could be found in a higher pH level with pH 7 degree. After menopause, pH again will rises and less glycosin secretion will be in place, and the flora returned to that found during birth again in the females. So this is how the microflora or microbiota or normal flora is different between in a human lifespan. During a human lifespan, you can see how the normal flora could be up and down and differ based on the factors, secretions, age, environmental factors. Based on that, the microbiota will be formulated. Yeast, for example, candida are occasionally found in the vagina. 10 to 30% of women could have a candida species infections. These sometimes increase and cause vaginitis. Urogenital flora. In the interior urethra of a human's, Staphylococcus epidermidis and enterococci and diphtheroids are found frequently. However, E. coli, Proteus, and Neisseria, non-pathogenic species we are talking about, are reported occasionally from 10 to 30% in this area. Because the normal flora residing in the urethra, care must be taken in clinically interpreting urine cultures. Urine samples, for example, may contain these organisms, as we say, E. coli, Proteus, Neisseria, for example. If a midstream clean catch specimen is not obtained, these bacteria could be disseminated. So we have to be careful during our clinical practices. Normal flora as well in the body, we are moving to conjunctival flora. Usually, approximately 70 to 49% of cultures of the samples are negative. So most of the time, no organisms in this area, no normal flora in this area. Lysozyme secreted in the tears may play a role in controlling the bacteria by interfering with their cell wall formation. Positive samples show bacteria like Crinobacteria, Neisseria, Moraxella are cultured. Moreover, Staphylococci and Streptococci are also present if the culture is positive and there is an infection. And recent report indicated that Haemophilus could be present too in 25% samples. So after understanding, finally, I don't know what's the time. Oh, wow, we have spoke for 1 hour. Still we have to go through some things. Sorry, I'm exceeding the time, guys. Are you having, do you enjoy the presentation? I want to move from the microflora, normal biota, to the microbial pathogenesis and how this pathogenesis could cause, how could this microbial cause a pathogenesis? Like, what is the relationship between these two things? I promise I'll give you a longer break, guys, after this. Microbial pathogenesis. So normal flora, this is pathogenesis. Microorganisms are in nature, and they are naturally present in and on humans. The terms used for those found on healthy surfaces is normal flora or common commensals. So normal flora, common commensals, microbiota are the same thing. Typical normal flora organisms vary by body site, as we say. The term colonization generally denotes the presence of microorganisms in the absence of symptoms or deep tissue invasion. Normal flora may be described as colonization in most cases. For example, E. coli colonization of the stool. هذه تعتبر normal flora, whereas potentially pathogenic organism may exist as a colonizer. Also, E. coli could cause an infection in the other side, but it is a colonization side, normal flora in the stool. Colonizing organisms such as Neisseria in the pharynx, Salmonella in the stool, methicillin-resistant Staphylococcus aureus in the nares, for example, and yeast in the genital tract. So these are examples of normal flora. which inhibiting parts of the body, may facilitate transmission to other or may lead to disease in the colonized individuals during disruptive situation. And this is what I was talking about. Whenever the normal flora is interrupted by disruptive situations such as antimicrobial treatment, invasive device, insertion, fresh wound cuts. In this case, this microbiome will be transient from normal flora to a pathogenic microorganism, which can cause a disease. Sources of microorganisms. Microorganisms also can come from a variety of sources. They can Exogenous organisms are those that come from outside of the person or the host. Those are the exogenous. Exogenous sources include other humans too, like herpes viruses. They come from another humans. Mycobacteria, tuberculosis as well. Food stuff like salmonella species, contaminated water resources. For example, insects with Lyme disease and malaria. Animals. For example, airborne sources, Legionella, for example, Legionella and Aspergillus. Those are airborne infections, and those can come from outside the human itself. So those considered as exogenous organisms are derived to, also endogenous whereas endogenous organisms are derived from the host itself, own microbial flora. For example, Staphylococcus aureus carried on the skin, or E. coli carried on the perineum. For some organisms, the distance is not clear-cut. Some pathogens may be acquired first as colonizers, or only to cause disease later. For example, they can colonize an area, but not cause a disease at the moment, but they can cause a disease later. They can be dominant, they can be latent, until they can cause disease later. Last slide, endogenous versus exogenous infections. Endogenous infection, when a person gets an infection caused by a pathogen that is already colonizing a part of their body. For example, Staph aureus in their nose. However, exogenous infections, when a person gets an infection from a germ that recently spread to them from another person or from a shared contaminated surface. This is the difference. I hope you get the full understanding of the microbiology, clinical microbiology. And if you have any question, just let me know. Or otherwise, we will go for 15 minutes break. Does colonization of the stool is not considered as a disease or as a colonization? Depends on the organism that you found in the stool. It depends on the organism. If this organism, like if you found in the stool E. coli, it's totally different than if you find Clostridium difficile. If you found in the stool Clostridium difficile, then the patient need to be isolated and contact patients. Need to use a single bathroom, and the bathroom should be clean with chlorine after the patient use, etc. Precautions. However, E. coli is fine. That's normal. So it depends on the organism that you find to be colonized in the stool. How can we study the organisms perfectly, and how can we memorize it? Well, it depends. Depend on your passion about microbiology. You have to read about it more. Just find a good book, basic book. If you are not microbiologist background, find the basic book about microbiology. Take a colorful one. I like colorful books, microbiology books, because they can show you pictures. You can see the organisms under the slides, under the microscopes, how they look like. So you will find it interesting to study. And if you are passionate about it, you will go through it well, I believe. Yeah, you are a nurse, as I thought. You have a book, because microbiology, guys, is basic. Microbiology, if you want to study and understand microbiology, it's a science which is basic, أساسي. Yes, there is a researches on it and continuous research on microorganisms, but the basics of microbiology is well written in books. I believe in books. How can we? Okay. Any other questions? Can you recommend one? Actually, I am a microbiologist, so أنا ما أقدر يا آلاء أقول لك one book. أنا قرأت books كثيرة بس long time ago. That's my bachelor degree, not now. But I believe you can search. You can search something you prefer. لأن البasic is basic. Any book, any basic popular book could help you, آلاء. Thank you, guys. I'll see you within, if خلود معانا, the moderator. I'll see you 9:30, guys. I'll see you 9:30. Okay, we will take 15 minutes break. Thank you very much. Good morning, everyone. أهلا. ما شاء الله. ما شاء الله. العدد مرة جميل، ما شاء الله. أهلا وسهلا. Hopefully, guys, by the end of our sessions today, you will get a better understanding about microbiology and the relevance of microbiology with infection control, infectious diseases. إن شاء الله تستفيدون وتستمتعون. Hopefully you will gain the knowledge and the fun at the same time. Right? أعتقد الرقم بدأ يرتفع. Most of you guys come back, so welcome. Thanks for coming on time. So next lecture will be about contamination, colonization, and clinical infection. And this lecture, I think it's another perspective from the previous lecture. Previous lecture is purely microbiology, pure microbiology. However, this one is relatively speaking about clinical infection and how does the microbiology interact with patients, with humans, causing contamination, colonization, clinical infection. Actually, these concepts, understanding the difference between them is crucial for any infection preventionist. And during my career, my long career in infection prevention field, actually these concepts are very confusing. For some reason, you feel that they are similar, you feel they are differ in situation than the other. You need to have a fully understanding about the basics and how could microorganism alter, and how the host mechanism defense will alter, and how those things interfere and interact with each others and cause any result of infection, colonization, or even just a contamination. So try to understand the outline topics today. It is more outlined than objectives. It's more overview than objectives 1 to 3 objective, because you will get a better understanding about microbiology history and scope. Microbes main types. We will talk about microbes individually, like bacteria, yeast, viruses, fungi, etc. Clinical microbiology and clinical side, not in a science side, but a clinical side in the hospital. Microbial pathogenicity and host response, how the host is responding to a microbe when it's altered from being a normal flora to a pathogen. Microbial ecology terms, which is usually infection, colonization, subclinical infection, for example, a carrier. You need to understand these definitions or terms so you can determine how risky is your patient. You can determine how could the risk come and where the risk comes from, and how to prevent and cut the chain of infection from that understanding. That's why it is important to understand the microbial ecology terms and microbiology foundation. Contamination, colonization, clinical infection. If you see, it is just at the end of the lecture, which is the topic aslan, but at the end, because you need to understand all these things before coming to a conclusion with what is contamination, colonization, and clinical, subclinical infections. And last but not least, general recommendation for all healthcare workers in the hospitals, no matter what kind, type, size your hospital is or your healthcare is center. You need to follow these recommendations in order to prevent contamination, colonization, and clinical infection from microbiological side. We are not going to discuss any, hand hygiene, PPEs, personal protective equipment, respiratory etiquette, cough etiquette, respiratory hygiene, etc. No, we are not going to talk about these interventions. We are going to talk about contamination, colonization, clinical infection, and relevant with microbiology. Okay? So microbiology history and scope. Basic definition in science, more clinical microbiology in science. Microbiology is the study of a tiny organisms which are too small to be seen by a naked eye, full stop. In science, microbiology is the study of tiny organisms which are too small to be seen in a naked eye. Clinical microbiology encompasses the study of pathogens such as bacteria, viruses, fungi, and parasites that cause disease or infection in humans. Issues of concern in clinical microbiology, بعض الissues اللي concern in clinical microbiology include the nature and epidemiology of the etiological agent and how they interact with the immune response, and their rapid diagnosis, and providing the information on the treatment or the best treatment could be, such as antimicrobial susceptibility tests. So clinical microbiology and laboratory main concerns is what we have said. Okay, the interaction of the immune system with the microorganism, rapid diagnosis, providing information on treatment, and how does this antimicrobial substance is susceptible to that microorganism. Fundamental knowledge of microorganisms and their identification and their significance And basic laboratory techniques provide the infection preventionists with a better understanding of pathogenic organisms. So, of course, as I said earlier, fundamental knowledge of microorganisms is crucial. And please find a place to get the knowledge from, for example, a book, a website. Try to read more about microorganisms if it's not your background, because this will aim and raise your practices as an infection preventionist much better if you have this understanding. Clinical laboratory methods that are used to evaluate the presence and or significance of microorganisms and methods to analyze the relatedness of microorganisms for epidemiological purposes. So this is the role of clinical microbiology and the laboratory, and how can these aspects help infection preventionists to prevent infection. Some key concepts that we will go through: certain qualities of microbes lie at the root of many infection prevention issues, including their extremely small size, rapid reductive rates, resistance to harsh environmental conditions. So certain characteristics of microbes can give the microbes a strong defense mechanism to the infection preventionists to fight them, because of their small size, rapid replication rate in a short time, and they're resistant to harsh environmental conditions. These all elements make it hard for the infection preventionists to prevent their growth. But with better understanding, we can do this. The clinical microbiology laboratory is an important partner in the practice of infection prevention, of course. And in this course, we have, I think tomorrow, one lecture is all about laboratory and infection control and how we get the best practice between each other. The clinical microbiology laboratory can provide clear information on microbes determined to be of clinical significance. So we can, from there, from the microbiology lab report, we can start our investigation, interaction, and prevention. Proper specimen collection and transport are integral to the recovery of failed microbiological information. Of course, transportation of the sample, how the sample has been collected with a proper aseptic technique could affect the result in the lab. And if we get a contaminated result from the lab or not clear result from the lab, then the whole process will be useless. Of course, practicing a simple sample collection best practice, transportation of the specimen also can affect the clarity of the result of the sample itself. The presence of microbes in a clinical specimen does not always indicate the presence of an infection, because in this case, we need to rely on several aspects, several things, like, let's say, first, the clinical status of the patient, and what does the doctor recommend, what does the doctor query about, what does the doctor suggestion, and is there any sign and symptoms on the patient, so we can say this is an infection or not. So the presence of a microbe in a clinical specimen does not always indicate the presence of an infection. We need to understand this concept. A variety of methods can be used to identify bacteria, fungi, and viruses, because by modern life, recent technology, there are a variety of methods to identify the bacteria, fungi, and viruses in a specimen. Antimicrobial susceptibility testing. This is really important for that, especially with regards bacteria and multi-drug resistant bacteria. is commonly used to assist in the selection of appropriate antimicrobial therapy to be used, because antimicrobial therapy is a huge topic that should have actually another course لحالة, because abusing the antibiotic therapy on the patients could lead to a worst case scenario and could lead to more drug-resistant bacteria in the world. And antimicrobial therapy should be very, very secure, tested, validated by the lab, by the infectious diseases consultant, by the clinical pharmacist. A huge team should involve during an antimicrobial therapy sessions. Monitoring resistance pattern is an important function, actually, of the clinical microbiology laboratory as well, because microbiology laboratory, they provide the infection control with reports about the resistance patterns in the hospital, and where we are now, and what we are heading to, what is the goal. We can also create a KPI with the laboratory people with regard monitoring resistant patterns in the facility that we are working in, controlling amount of resistant patterns, and keep an eye on those patterns is very, very important role in infection control and laboratory. The clinical microbiology laboratory is an important collaborative partner during outbreaks investigations as well, and situations required environmental sampling. They are a very collaborative partner, actually, in this kind of situations and emergency cases, of course. So microbes, main types in general. The field of microbiology includes the study of bacteria, fungi, molds and yeast, protozoa, viruses, and algae. Infection preventionists are likely to encounter most of these microbes types in the course of their practice, with the exception of algae. Algae مو موجودة بالhospitals. Structures and identifications of bacteria species is described first in this coming slide, following by fungi and viruses. So we will discuss bacteria today and fungi and viruses, their characteristics, their structure, their arrangement, and how they replicate and how they invade, etc. So you will get a better understanding. First, bacteria classification. There are at least 17 groups of bacteria according to one classification scheme that is based in a large part of nucleotide sequence of RNA. All of these groups have a similar cell structure that is known as prokaryotics. Prokaryotic cell structure is microscopically characterized by lack of a visible nucleus, by the lack of a membrane nuclear envelope, and by a lack of membrane-bound internal structures such as mitochondria. So those are prokaryotic, and bacteria fills in there. Cell structure and arrangement. Bacteria are very small and a single-cell organisms. They contain a single long circular nucleus of double stranded DNA. This bacterial chromosome is not surrounded by a nuclear envelope and is attached to the plasma membrane. In addition to the bacteria chromosome, bacteria often contain a small circular double-stranded DNA molecules called plasmid. So the bacteria has DNA double-stranded plasmid in it, and this plasmid contain genetic information about this bacteria. It holds the whole genome, the whole genetic characteristic of this bacteria. Although plasmid are not necessarily for cell survival, in a normal environment, they may carry genes for activities. So the plasmid carry the genes for certain activities, such as antibiotic resistance, production of toxins, if the bacteria could produce a toxin, just like gram-negative bacteria, and the synthesis of enzymes, the ability to synthesize the enzymes. Those codes are integrated inside the plasmid of the bacteria. Therefore, the bacteria has the information inside its plasmid. The information of the statistics، إيش القدرات الخصائص تبعت البكتيريا، سواء if it's antibiotic resistant, if this bacteria could produce toxins, if this bacteria even could produce spores, and if this bacteria can synthesize of any enzymes. Okay. Still in the cell structure and arrangement, plasmid can be transferred from one bacterium to another. Of course, because it can handle the genetic information within it. So from one bacterium to another, and genes may move. Genes may move from plasmid to a chromosome, to another chromosome. These genes are called transposable genetic elements. Bacteria also contains ribosomes that function as the site of protein synthesis. So البكتيريا عندها ribosomes. Ribosomes is the function as the site of protein synthesis in the bacteria cell. Okay, the plasma membrane encloses the cytoplasm of the cell and provides selective nutrients to be enter and come out from the cell. هذا البلازما membrane has an axis entrance to the cell, depends on the nutrients need to the cell. The cell wall of a bacteria is a complex topic صراحة. Semi-rigid structure responsible for the shape of the cell. And here I would like to say that gram-negative bacteria is different than gram-positive bacteria from the cell wall part. من السيل وول، الجدار الخلوي للبكتيريا الموجبة لجرام يختلف عن الجدار الخلوي للبكتيريا السالبة لجرام بصبغة جرام. فالسيل wall is a very important characteristic in the bacteria because it has the authority to identify two types, two major classes of bacteria strains, which is gram-positive and gram-negative bacteria. It surrounds the underlying. السيل wall يحيط بكل شيء من الداخل. It surrounds the underlying fragile plasma لأن البلازما membrane is fragile, and protect the cell أكيد, and the interior of the cell is protected by the cell wall, of course, from the environment. The cell wall is made of macromolecular network called peptidoglycan. So peptidoglycan is a core component actually, in a bacteria cell wall structure. Again, in most gram-positive bacteria, the cell wall consists of many layers of peptidoglycan forming a thick, rigid structure. In contrast, however, gram-negative bacteria cell wall contain only one or few layers of peptidoglycan. وهنا الفرق بين البكتيريتين. Gram-negative cells possess an outer membrane that is composed of lipoproteins, polysaccharides. هذا الكومبوننت is found in gram-negative bacteria, the polysaccharides, and phospholipids. This outer membrane helps some organisms evade phagocytosis and provides a barrier to certain antibiotics. So هنا في characteristics للحماية لنفس البكتيريا by avoiding phagocytosis من immune system and providing a barrier to a certain antibiotics therapy, and converse properties of virulence to the bacteria to be to produce endotoxins. So these characteristics related to the gram-negative bacteria, which can produce endotoxins, which can evade phagocytosis, which can provide a barrier to the certain antibiotics, are harsh characteristics actually that gram-negative bacteria owns, in fact. Glycis is a general term used for widely varying chemical substance that's surrounding the cell. Okay, and if this substance is organized and firmly attached to the cell wall, it is referred to as a capsule. So الكبسول هو الجلايكوسين. The capsule of the bacteria is the glycosine material. Capsules may contribute to bacterial virulence, of course, and may offer protection from phagocytosis as well. So whenever bacteria has a capsule characteristic, meaning this bacteria is virulent more and can tolerate defense mechanisms and can offer herself a protection from phagocytosis, etc. If the glycosine is unorganized on the cell wall and only loosely attached to the cell wall, مش firmly attached, it is called a slime layer. Sometimes you can see a bacterial growth that is slimy. When you touch it, you feel it's slimy. And this is a glyco layer, which is loosely attached to the cell wall of the bacteria, not firm. Some bacteria have a flagella that are long, appendix, which is helping the cell to move. So the flagella is a structure attached in the bacterial cell, which can help and aids bacteria to move. توني انتبه للأسئلة. Can you hear me, guys? Oh, thank you. Okay. Many gram-negative bacteria possess hair-like appendages that are used for attachment rather than motility. يعني الزوائد اللي تجي على خلية البكتيريا مو بشرط انها just for movement. It's not only for movement purposes. Sometimes it helps the bacteria to attach, okay? To attach to the host cell more, firmly attached to the host cell so they can invade the host cell. These are divided into two types. Singular or double البلاي. These attachments for mortality and attachment could be singular and could be double pilies. They are enable the bacteria cell to adhere to the surfaces, including other cells, whereas pili join bacteria cells in preparation for the transfer of DNA from one cell to another. Cell shape and arrangement are usually microscopically observed for many species. It's observed very clear under the microscope, the shapes and the arrangement of the cells. When essential nutrients are non-available, certain gram-positive bacteria such as Clostridium bacillus form a structure known as endospores. Endospores are composed of nuclear material and protein. So the endospores contains nuclear material and protein as well. So it has the whole data of the mother cell of the bacteria, but it is an endospore form. Endospores are remarkable for their ability to survive in water heated to above of boiling temperature, and for their ability to survive extremely in drying weather, and their ability also to survive exposure even if they are exposure to some toxic chemicals. So the endospores forms are very crucial, very harmful, very intense, and they need special care, special disinfection, cleaning process, hygiene, etc. When growth conditions permit and the endospores transfer from being an endospore into more typical bacteria known as vegetative form. A vegetative form is the basic form of the bacteria, but when the environment is not suitable for growth, there is no enough nutrition, there is no enough humid, there is no enough factors of growth of this bacteria, or the environment is not in favor with the bacteria, the vegetative form will be transferred to be an endospores, and vice versa. When the environment is suitable, the endospores become a vegetative form. An example of this is seen with Clostridium difficile associated diarrhea. When the patient has active diarrhea, the feces contain both the vegetative and the endospore forms of the bacterium. That's why we need to adhere to contact caution, keep a patient in a single toilet, clean the toilet with chlorine products, and hand rubbing is not enough with alcohol hand rub, but hand washing with soap and water is the best practice of hand hygiene with Clostridium difficile associated diarrhea and infections. Once shed from the body, the vegetative bacteria forms endospores that are not easily killed by high temperature detergents or disinfectants that are used to clean equipment and facility. When endospores encounter a favorable environment, such as the interior of a human intestine of one, they can transform into vegetative cells that are capable of reproduction again. Okay. Bacteria normally reproduce by binary fission, with one cell dividing into two cells. So we have ways of reproduction, how bacteria reproduce. First way is bacteria normally reproduce by binary fission, with one cell divided into two cells. The rate of division can vary from slow, like in the case of Mycobacterium tuberculosis. They replicate every 12 to 24 hours. To a rapid replication, just like the case with E. coli cells, for example, they may divide it every 20 minutes under ideal environmental conditions. So the division and multiplication of bacteria are differ from strain to strain. For Mycobacterium tuberculosis, it takes long time than E. coli, for example. The first step in division is the cell of the bacteria getting elongation, يعني استطالة, and replicate of chromosal DNA. The DNA inside the mother cell will be replicated and the cell will get a longer shape, okay? The cell wall and the cell membrane begin to grow inwards, inside the cell, near the middle of the cell, eventually meeting in the middle to form a cross wall in between. Thus, two individual cells will be formed at the end, اللي هو الانشطار الخلوي أو الانقسام الخلوي بالعربي. These daughter cells are essentially identical to the parent cell. أول انقسام يصير identical يشبه الparent cell. First binary vision happened. The outcome cells will be identical, typical, same as the parent cells, without any modification, without any changes, okay? The changes will be later, but the first one is the same. Bacteria are known to be obtained in new combinations of genes in several ways. As I said, after the daughter cells has been become from the parent cells, bacteria could be multiply in a different ways, and they love to integrate with other DNAs, other components, so they get more challenges. They shape themselves to have a strong character from each thing they get merged with, because in this case the bacteria will become more harsh, more independent, and more virulent, in fact. So there are, I think, five steps to get bacteria in combination with genes and other genes in several ways. First, transformation. Transformation phase is occur when a naked DNA in the environment, naked كذا DNA in the environment, يعني after bacteria تحللت or whatever DNA naked in the environment, possibly from a dead bacteria, enters another bacterium. هذا transformation يسمونه. The transferring DNA either may be incorporated into existing chromosomal DNA or transferred DNA plasmid كامل. يعني ممكن يكون جزء من البلازميد, can be جزء part from the plasmid or part of the DNA found in the environment, or it could be the whole DNA integrated. Second is conjugation. Occur when all of or part of a plasmid is transferred from a donor to a recipient cell. The cell must be in direct contact, and transfer occur via the sex valence. Conjunction can occur between widely spread species. And many species, less species, that's fine. They can transfer their characteristics between them, leading to a rapid dissemination of genetic information. ومن هنا it comes from there the antibiotic resistance genomes. Okay. Third is transduction occurs when a bacterial DNA is transferred from a donor cell to a recipient cell via a virus capable of infecting this bacteria. Also, this can reflect on the bacterial genetic material, bacterial characteristics, bacterial virulence, etc. So from these aspects, the bacteria can get a combination of genes and survive. Yeah, so they are three ways of getting a bacterial genetic exchange, if I could say. Right. Fungi. Fungi are eukaryotic, مش prokaryotic. prokaryotic غير حقيقية النواة. Eukaryotic حقيقية النواة. So fungi are eukaryotic organisms. The term eukaryotic indicates that they have a nucleus that is visible with a light microscope and that is surrounded by nuclear envelope. عندهم nuclear, عندهم envelope, clear structure under the microscope. ما نحتاج صبغة عشان يبينون ولا شيء. The nucleus is typically spherical or ovoid, about 6 nanometer in diameters. This is the nucleus. Relative size of fungi compared to bacteria can be appreciated by calculating that about 200 spheres the size of streptococci bacteria cell could be fit into a nucleus of typical fungi cell. And a fungi is quite bigger than the bacterial cell. Fungi cell is quite bigger than bacterial cell. Fungi get nutrients from organic materials. Usually they feed themselves by organic materials. Fungi cell walls contain a chemical substance known as chitin. Some fungi are well adapted human microorganisms such as Candida albicans. مش Candida aureus, Candida albicans. They are, as I said previously, one of the normal microflora in the body. Most however, are accidental pathogens. Most of them are accidental pathogens, unfortunately, that humans acquired through contact with decaying organic material or airborne spores in the environment, especially during construction, renovation. As you know, during construction, renovation, the most thing we care about is aspergillus from the air conditioning to be developed during the shut on, shut off of the ACs, the filters of the air conditioning system. So typically, fungi may be divided based on their appearance of the organism into two groups, yeast and molds. So the fungi are divided based on their appearance into yeasts and molds. Yeasts are single cells, microscopic organisms. In culture, yeast usually form a smooth, creamy colonies. You can see them on the plate of the agar. They have a single nucleus with a nuclear membrane and contain organelles. Okay, they typically reproduce by a process of bonding, اللي هي التبرع. In this process, a parent cell forms a bud on its outer surface. As the bud being long, the parent cell nucleus divides, just like الانشطار البكتيري, divides and one nucleus migrates into the bud. So eventually the cell wall closes between the parent cell and the bud comes out and the bud breaks free. Some yeast reproduce through the process of fission, similar to the bacterial reproduction as well. Common pathogenic yeasts include Candida species, which can cause mucositis, for example, vaginitis, dermatitis, or systemic dissemination also, and Cryptococcus infections, which can cause meningitis, pneumonia, in compromised hosts. So again here, compromised hosts, immune deficiency host could be quite affected by the pathogens that are present on the body. Molds. Please, guys, don't write your excuses on the questions and answers because I guess I thought إنه عندكم أسئلة, but it's not, it's just a chat. So keep question and answer section, please, just for questions and answers, so we can make the conversation much clearer. Thank you. Molds. Molds consist, اللي هو الجزء الثاني من الفنجاي. حكينا عن اليست and now المولds. المولds consist of long, branching filaments of cells called hypha. Okay, a tangled mass of hyphas visible to the naked eye is called mycelium. Aerial mycelium gives a mold a fuzzy and woolly appearance in a culture. كذا شكلها زي الصوف المتناثر اللي هو الhyphas having المolds. The hypha's physical characteristics such as shape, pigmentation, and the presence of root-like structure called rhizoid, okay, are used to differentiate and identify molds in laboratory. بناء على هذه المعطيات. Some molds reproduce asexually by fragmentation of their hyphas. ينكسر الهايفا لنصين ويصير فيه production بالfragmentation method. Additionally, بالإضافة إلى asexually method, additionally, molds may replicate sexually and asexually by forming spores. يعني the spores could be produced من المولds with a sexual and non-sexual method. However, المولد نفسها بدون spores, without spores, it replicate by asexual method اللي هي fragmentation للhyphas. So asexual spore are formed by the hypha of one organism. When these spores germinate, they become organisms identical to their parent cell. Okay, when they germinate. But the sexual spores result from a fusion of nuclei from two opposite matting strains of the same species of the fungus. Organisms that grow from sexual spores have characteristics of both parents' strains, of course. لأنه أخذوا صفات من هنا ومن هنا. Common opportunistic pathogenic molds include Aspergillus. And Aspergillus species can cause invasive pulmonary aspergillosis. And agents of mucormycosis as well. Okay, هذه برضو mold. Next are viruses. Viruses are not considered to be cells. أعتقد هذه المعلومة فاجأت كثير non-microbiologist people. قولوا لي. Yeah? فاجأتكم? Did you surprise because viruses are not considered as to be a cell? Absolutely, Abdullah. طيب, bravo. طيب. Therefore, they are not classified as either eukaryotic or prokaryotic. They are not classified على هذا الأساس لأنهم أصلاً مش خلايا. Okay? They are obligate intracellular parasites. هم كذا. Which means that they grow and replicate within living cells للhost طبعاً and are depending on the cell synthesis and metabolic machinery. They cannot live alone. They have to have a host, and they use the host energy, nutrient, mechanisms, synthesis to live inside. So viruses are too small to be seen with a light microscope, regular العادي, although they may be visualized with an electron microscope. So viruses could not be seen by light microscope العادي. They can be detected or seen by the electron microscope. Viruses particles contain nucleic acid, either RNA or DNA, surrounded by protein, and in some cases, other components such as membrane-like envelope. Some viruses have envelopes and some have not. Originally, viruses were classified by their type of host. عادة, they classified by the type of host and the type of disease they can cause. For example, human polyvirus. هذا يعني يتسمى الفيروس بناء على الhost they can invade and the type of disease they can cause. It can be either this or that. With the presence of genetic testing, الجديد, recently in the world, viruses are now classified in families and genera based on a genome typing, RNA or DNA, and the number of strands in the genome, either a double strand, DS, or a single strand, SS. Also based on their morphology, they classified, and their presence or absence of an envelope, if they have envelope or none. So those are the characteristics that viruses are classified by. Viruses reproduction process. Now, outside the host cell, the virus's particle is known as a viron. It is metabolically inert and does not grow or multiply until it enters a living cell. Viruses in general replicate in a similar fashion, which occur in 5 steps. So the viruses needs 5 steps to be replicating. First step is the attachment, لأنه هو متطفل فيحتاج إنه يعمل attachment أول عشان يتكاثر. Okay, so first step is attachment because it's an obligate organism, so it needs to attach to a host cell so it can replicate. The viron اللي هو في الانفايرمنت الحالة كان اسمه viron مو فيروس لأنه في الحالة environment يعني زي اللي latent لسه ما عاش ما هو حي enough لأنه ما بعد غذاء الخلية العائلة. So the viron attached to a complementary receptor site on the host cell. Virus attach is a specific. لازم نعرف إن الفيروس is very specific microorganism. It attached in certain places, not in any place. For example, Epstein-Barr virus attached to a receptor on a B lymphocytes. B lymphocytes هذا خلايا مناعية immunocells. The virus, this virus اللي هو Epstein-Barr virus is only attached to this specific area in the B lymphocyte receptor. The first virus needs to be attached. Okay. Second, penetration. The viron enters the host cell through a process called endocytosis. Endocytosis. هذه في penetration stage, which means that the host cell engulfs. The host cell تبتلع الانفايرون هذا الفيرون. The virus, rather like it, brings food molecules into the cell على أساس كأنه شيء يعني تتغذى عليه. It's just like looks like a food molecules to the cell, so the cell, the host cell, engulf the virus inside the cell itself. Number 3, when the virus cell is engulfed and penetrated the host cell, number 3, the virus cell will be replicated, will have the ability to replicate viral DNA or RNA على حسب نوع الفيروس, directs the host cell, خلاص يبدأ يقود الآن, to begin synthesis of viral components, viral replications, uses the host cell ribosomes لمصلحته الشخصية, uses the energy sources, uses the amino acids to produce these components. So يبدأ يشغل, it start to use all the mechanism or nutrients and living growing mechanisms inside the host cell to its benefit, to its own benefit. Number 4, maturation. حين الخلية تعمل لصالح الفيروس لكن نحتاج الفيروس يحتاج يكون mature enough so it can release, بعد كذا يعمل عملية إنه يطلع للحياة ويستقل عن هذه الخلية العائلة. So maturation level is the viral components essentially assemble into a viral particles spontaneously forming a daughter virus. يبدأ يتكاثر ويقوى, يتكاثر وينضج جوا الخلية العائل. Number 5, release. The host cell lyses. تبدأ خلية العائل تنفجر أو تتحلل. So the host cell lyses, or في خيار ثاني إنه the virus buds through the, يتبرعم من الخلية وهي موجودة, يعني ما يفجرها, يطلع منها بعد ما تكاثر وصار قوي وماتيور, يطلع منها. The virus buds through the cell wall عادي, and the daughter virons are released من السل, بس السل أكيد تكون ضعيفة بعدهم أكيد. Some viruses lay dormant in the host cell, يكونون ما يطلعون, ما يتبرعمون, ما تسوي release. They still hold in the host cell for months and some for years. And after this latent period, new virons form بزيادة and cause damage to the host cell at the end, of course. Virus culture. علشان نزرع الفيروس في اللاب. Virus cultures traditionally requires a specialized media containing antimicrobial, antifungal agents prepared in a plastic or glass tube and flasks. Because ممكن إنه لو كان في بكتيريا أو fungal ما يقدر the virus cannot be exist. So we need to have antibacterial to the media, antifungal to the media, so the viral could be stronger and multiply in this media. Clinical specimens are cultured on a group of different mammalian cell culture lines, depending on the agent suspected clinically, depending on the suspected agent suspected clinically by the physician. Growth is viewed microscopically and is identified through changes in the host cell rather than as original viral colonies. Although tissue culture are still considered to be the gold standard for viral culturing, but many viral infections are diagnosed through other rapid methods, يعني مو شرط tissue culture. Advantages of viral cultures include sensitivity, detection of many types of viruses, adaptability to the viral variation, and the options of susceptibility testing. All these elements are advantages in a viral cultures. Next, بعد الفيروسز we are going to talk about mycobacteria. Mycobacteria identification. Mycobacteria require the special culture techniques to be isolated. In general, specimens undergo procedures to kill commensal bacteria that may be present. Once the specimen has been processed, it is planted on a special media and incubated for 4 to 6 weeks. هذا عشان نسوي mycobacteria culture. After the organism grows, further testing must be conducted to identify the species, tuberculosis and other mycobacteria species. Because conventional culture techniques may take weeks to recover a mycobacteria growth, more rapid techniques have been developed recently. For example, PCR methods. PCR جدا مفيد في موضوع الميكوبكتيريا nowadays. لأن الميكوبكتيريا takes long time to grow, as we said. ما هي زي E. coli وغيرها. Not like E. coli and other bacteria. So PCR method in this case have been developed. In this method, the genetic chromosal parts can be detected by DNA probes للميكوبكتيريا. DNA probes usually are used after the mycobacteria have been isolated from the culture. Okay. Clinically, خلصنا جزئية الحين الميكروبيولوجي. Okay, هذا كله عشان عفوا all this thing is just to understand how we can enter to colonization, contamination, and clinical, subclinical infections. So after reviewing all this information from a microbiology perspective, clinically, the presence and identification of organisms in a clinical specimen may be an evidence of infection. It might be an evidence of infection, it might be not. The primary goal of a clinical microbiology are to identify the presence of a pathogenic organism in a tissue or a body fluid or extraction, secretions, and to identify those pathogens to species level based on morphological and biochemical properties. Additional goals are to predict response to antimicrobial therapy and assist in the epidemiological investigation. So من هنا برضو مهم الclinical microbiology. Often the first step to identify microbes is preparation of the slide, صح? To examine the clinical sample microbiologically. Another necessary procedure in bacteria identification is preparation of pure culture. علشان نقول دي البكتيريا, we should isolate a pure culture, bacteria in a pure culture. Note that not all microbes can be visualized with a microscope, nor can all be cultured in the hospital laboratories. Some samples need to be sent to advanced laboratories outside the hospital. Microbial pathogenicity and the host response. Infection, لأن الحين بندخل, we will talk later يعني after a while about infection زي ما قلنا contamination and colonization. Infection results when an imbalance occurs between a mechanism that microorganisms employ to induce an infection and the complex psychological response system that are employed by the host to prevent such an infection. من هنا تنشأ الانفكشن. This is where the infection can come from. Infection can be distinguished from colonization, which is when a microorganism occupies an ecologic niche in a host, grows, but does not overcome a host resistant or cause invasive disease. هذا الفرق بين الcolonization والinfection. In past decades, increases in microbial virulence اللي لاحظناه في past decades induces by changes in exotoxin production of the microorganisms or surfaces antigen construction have led to a better understanding of determination of the virulence in microorganisms. We have also come to understanding also the interaction within the immune system that serve the foster integration of the host defense, and how human immunodeficiency virus, the immune altering prototyping infection of the 20th centuries modified and defeats those defenses. Additionally, we are now able to distinguish بوضوح those elements of natural defense and sometimes referred to innate immune system, which are activated by a cell injury or death, etc. And adaptive immunity, اللي هو cellular ولا hormonal, that can be enhanced and have a property of immunological memory. In many circumstances, we can enhance or support a state of strong adaptive immune and terminate or prevent the infection. In many circumstances, we can prevent infection, of course. Understanding this defenses mechanism is essential, of course, to the design and implementation of effective infection prevention and control programs in healthcare settings. Antimicrobial therapy seeks to suppress bacteriostatic. So antimicrobial therapy, either doing a suppress اللي هو bacteriostatic or killing the bacteria اللي هو bactericidal للmicroorganism by exploiting biochemistry reactions unique to the pathogenic microbe. Ideally, this should be accomplished using the simplest agent with the minimum toxicity or to the patient. يعني the dose of the dose of antibiotic is quite important. Concentration is quite important. Type of the antibiotic is also quite important to understand the antimicrobial susceptibility test. Okay. Originally, the purpose of susceptibility test, sometimes referred to as sensitivity testing, was to determine whether the organism isolated was able to resist the effects of the therapeutic agent chosen for treatment for this patient. Also, in antimicrobial susceptibility testing, we need to know how process have evolved. There are antimicrobial susceptibility testing they call AST methods available that directly measure the efficiency or the activity of one or more antimicrobial agent against the bacterial isolates, directly detect the presence of a specific resistance mechanism in a bacteria isolate, and measure the complex antimicrobial organism interaction at the same time. The type and extent of the AST, اللي هو antimicrobial susceptibility testing, conducted depend on the organism isolated, the source of the culture from the body side, for example, the available antimicrobial agents in the laboratory, and finally, the typical susceptibility pattern in the guidelines, written in the guidelines. Several methods may be used for susceptibility testing also. منها, one of them is the disk diffusion method to consider or to understand antimicrobial susceptibility testing. Once a potential pathogen has been isolated by a culture, a standardized suspension of bacteria is spread in a lone version of a agar plate, and a paper disc soaked with a standard amount of antibiotic placed onto the agar surface, and the agar plate will be there incubated for overnight. And during the incubation time, the antibiotic diffuses out into the agar, of course, resulting in a decreasing concentrating of the agent as it moves far from the disc itself. Organism growth is either inhibited by the concentration of the antibiotic in the agar plate or not. That area in which the concentration of the antibiotic prohibits the growth of the organism is called the zone of inhibition. Disc division antimicrobial susceptibility test can, this measurement of the zone size have been standardized, of course, for each antibiotic, for each pathogen interaction, and are used to interpret the effectiveness of the antibiotic or class of the antibiotic with the specific isolate microorganism. Once the zone of inhibition has been measured, of course with a ruler in millimeters, it is compared with the CLSI, the Clinical Laboratory Standard Institute's guidelines for interpretation. The disc diffusion is reported in one of three ways. At the end of the disc division test, the result should appear from the laboratory in a report to the infection control, to the patient file, as one of the three answers, certain answers, inshallah. One or two or three. One, susceptible, meaning the organism is susceptible to be die or killed by this antibiotic, by this concentration, by this class, etc. So first is susceptible, indicating that the antimicrobial agent may be effective against the identified pathogen. Number two is intermediate result, and this is a shadow area. It's not very clear. It's confusing while starting the antimicrobial therapy, and it's not preferable from infection control side. We would like to see either susceptible or resistant. However, some results come back with intermediate reaction. Intermediate indicates that the antimicrobial agent may be less effective than the antimicrobial agent with a susceptible result. Of course, it's not susceptible, it's intermediate. So, أكيد يختلف. That the organism may become resistant. The organism may become resistant. يعني بس ما راح يصير susceptible. The intermediate will not become susceptible, may become resistant to the substance. Therefore, intermediate, it's look like resistant result. Yeah, if you agree with me. Or an antimicrobial agent may potentially be used if a high drug concentration is administered. maybe we need to add more concentration of this antibiotic to get a susceptible result rather than intermediate. Okay. Number 3 is a resistant result, and a resistant is a result indicating that the antimicrobial agent should not be used for therapy in this case, meaning the microorganism is resistant to this antibiotic. Try another class of antibiotic. Broth dilution antimicrobial susceptibility testing. There is another type of testing as well, susceptibility testing, which is a broth dilution. Broth dilution test method are used to determine the least amount of antibiotic necessary to inhibit the growth of the organism, or the minimum inhibitory concentration. We call it, in short, MIC. And the MIC is important to avoid future multi-drug resistant patterns, to avoid excessive use of antibiotic, to avoid harming the patient body, to avoid harming normal flora of the patient body parts, etc. So MIC is very important concept, needs to be implemented during using an antimicrobial therapy. This method uses replicate inoculation of the standardized substitution of the bacteria in a broth into a series of microwalls containing antibiotic in decreasing concentration expressed in microorganism per milliliter. Okay. After a period of incubation of microwalls, the walls are examined after an incubation period for bacterial growth. Okay, but each wall من هذه الوالز الجدران اللي موجودة في الشريحة has different concentration of the antibiotic used. So after an incubation, seen as a turbidity, we see bacterial growth في الوالز هذه كعكارة, turbidity. The first wall in the series that shows no bacterial growth, no turbidity, contains the minimum inhibitory concentration of the antibiotic that is effective against the organism being tested. هنا نقول هذا the best IMC for this case. Broth dilution test results are reported in microorganisms per milliliter. The reading is compared with set breakpoints determined by CLSI, of course, for interpretation and sensitive, intermediate, or resistant levels. Finally, contamination, colonization, clinical infection. بعد ما فهمنا كل هذه الأشياء, after the whole understanding for what I have said since morning, microbial ecology terms. First, we need to understand the terms, and then from there we can determine what are the types of our patients and how can we protect them and how can we consider them as a risk factor or no. Colonization, when a microbe is found on an or on or in the body, but does not cause symptoms or disease. Finding the microbe multiple times over time could represent a persistent colonization, but no clinical expression, no immune response. This is the colonization. This is how colonization looks like. Okay, the relationship between the host and the agent for colonization type in which the immune response is difficult to detect. في host وفي agent في يعني عامل يغزو أو مثلا بكتيريا ولا فنجل ولا فيروس, بس difficult to detect an immune response في colonization stage. Okay. Next is the infection, when a microbe such as bacteria, fungus, cause the disease in a living organism, in any host, people, animal, it could be. هذه الانفection. Contamination is the presence of microorganisms on a body, surface, or an inanimate object. بس presence. Only the presence of microorganisms on a body surface considered as a contamination. The carrier is an individual which is colonized with microorganisms. Subclinical infection, the presence of microorganisms and interaction between the host and the organism is just started, but a subclinical response only is there, and there is a little immune response. هنا subclinical infection. So in this picture, guys, this is very nice one actually. It can give you a better understanding between microorganism and immunity perspective and infection control perspective too. So the picture starts from the contamination. If we imagine this is the surface of any host, a surface, it could be stomach, it could be skin, it could be eye, it could be lungs, it could be any surface, okay? And there is a presence of microorganism on this surface. So this means that we are in the contamination time, in the contamination phase or stage, right? So after the contamination, if this persists and replicate, if the environment is suitable for these microorganisms in terms of humid, temperature, etc., and the microorganisms start to replicate, just replicate without invasion, listen, this is called colonization. They are here, they can cause an infection, but they are still in the stage of colonization. Critical colonization is multiplying and starting an invasion. And هنا عندنا subclinical infection started. And the infection will be local, considered to be a local level infection because it's just started in this designated area. It's not systemic yet, it's just controlled located area of invasion. After a clinical colonization, when it's happened, they become more and more, and this barrier will become weaken more. So the microorganisms will replicate, the barrier will be intact, and the infection become systemic. And here we can have virulence, erythema, inflammation. This is as a skin, for example, heat. And here we can say it's an infection stage. So to prevent infection, to prevent subclinical infection, to prevent colonization, we need to do proper decontamination. Proper decontamination. Proper practicing of infection control measures. Hand hygiene, environmental hygiene, personal protective equipment, personal hygiene for the patient, for the staff, etc. So whenever we do decontamination, the possibility to get infection is low. Sometimes we cannot avoid colonization. Sometimes people just born with it. Sometimes they came from a geographic region with it. You cannot decolonize. Sometimes you cannot decolonize, but always you can decontaminate. Always you can decontaminate. Infection versus colonization. More depth. Colonization, the presence of microorganism on the host, as we said, with the growth and multiplication, but without tissue invasion or damage or clinical signs and symptoms. Understanding the concept, هذا concept is essential in the planning and implementation of epidemiological studies and healthcare infection prevention and control programs. Confusion, colonization with infection, مرة غلط, can lead to suppress association that may lead to expansive, ineffective measures, time-consuming, wasting for the resources. So we need to understand, not confused between colonization and infection concepts. Colonization may become infection when changes happened in the host. As we said, when the host become immunocompromised, immunosuppressed, bad lifestyle habits, bad nutrition, malnutrition. Yes, in this case, colonization can become an infection. Does colonization protect the host؟ هل الcolonization sometimes تكون شيء كويس؟ Well, as we said earlier, normal flora protects against infectious diseases originating at mucous membrane, for example. There are several mechanisms for protection, مثل non-specific stimulation of immune responsiveness and specific cross-reactive immunization, زي vaccination, etc., and competitive bacterial interventions or interference, bacterial growth interference من colonization ومن الnormal microbiota. Shifts in colonization microbiota. It could be happened, as we say earlier, general shift toward gram-negative flora in hospitals. يعني hospitals recently خاصة in long-term care facilities having gram-negative flora as a normal flora في facility, وهذا شيء it's not nice, it's not something we wanted to be happened. And also in other healthcare facilities. And this is like a risk factor. Modification of the skin environment due to the skin changes is still poorly understanding, لكنها contraindicated, لكنها correlevant with the issue. Invasive procedures provides portal, أكيد, an easy entrance portal of entry to the different flora, to the body and invade. Antibiotic therapy in a study shows that the patient of ampicillin long-term antibiotic, 90% colonized by ampicillin-resistant enterobacteria. By time, they become colonized with resistant strains due to antibiotic therapy, even if this was a normal flora pathogen. Here is the sites of flora colonization. If we can see, the upper part of the body has the same in general, Streptococcus, Staphylococcus, Moraxella catarrhalis, for example, the upper intestinal and lower intestinal. Probably the upper part of the body has the same, and the lower part has the similar normal flora as well. What is the carrier? A carrier is an individual that harbors a specific microorganism in the absence of clinical infection, disease, and serve as a potential source of infection. يعني قنبلة موقوتة, a carrier. A carrier may be an individual who is colonized. يعني مهم إنكم تعرفون هذه الأشياء. He could be an infected and asymptomatic, يعني infected but asymptomatic. He could be in an incubation period before the disease is showing. Or he could be convalescent from acute disease at the end of a stage of an acute disease, for example. And the carrier status may be short, may be long, it depends. برضه على depend also on the human immune response, the host mechanism, lifestyle, etc. Okay? The infection is the replication of the organism in the host tissue, which may cause a disease. Infection defines the entrance and development of the infectious agent in a human or animal body, whether or not develop a disease. Caused by an infectious agent, all micro or macro organisms capable of producing an infection or an infectious disease. Infectious disease is the illness caused by a specific infectious agent or its toxic products. يعني ممكن مو بسبب البكتيريا لكن التوكسيك اللي يطلع من البكتيريا. Might be like this, an infectious disease that results from transmission of that agent or its product from an infected person or animal or reservoir to a susceptible host. Clinical infection may result in a sign and symptoms. Some of these may be less obvious or very minor. Sometimes, I mean, the clinical infection is not, the signs symptoms is not really showing, if they are very minor, but the infection is there. And at the end of the spectrum, sometimes at the end of the spectrum of infection, the individual with no sign, no symptoms, who has asymptomatic infection or subclinical infection, ممكن تكون at the end of the infection spectrum. Asymptomatic infection does not mean that all is quiet, all is good. No, it may cover some very active process happening inside the patient as the asymptomatic phase of the مثلا HIV infection, or for example tuberculosis infection, or for example hepatitis B carrier state. All those are asymptomatic infection at the beginning due to their long incubation period. So they are not really quiet as they show the quietness. Finally, general recommendations that if we implement, I believe, if we decontaminate, if we communicate well with the laboratory, if we have a better understanding about microorganisms and normal flora and how they interact with the human body, we can come up with a better practices, better implementations, better understanding. So first, make infections and MDRO prevention and control and organizational patient safety priority. This is number one. Keep it in your KPIs, monthly reports, quarterly reports. Keep reminders inside the hospitals about making disinfections and MDROs prevention as an organizational patient safety priority, not only related to infection control department, just that, no, it should be the goal of each and single department. Number two, implement a system to communicate properly with information about reportable infections and MDROs to the state public health authorities. This will keep up-to-date rates, national rates to be more realistic, and whenever you need a support from these public health authorities, you will gain a best knowledge, a best support to your institution, and never be shy, never be silent whenever there is a risk in your institution. Just show your plan, show your dignity, try to work better and solve things all out. Number three, support participation of the facility to combat emerging or growing MDROs, problems or infections, for example. Number four, provide education, and this is really important, very important side. Provide education and training on risks and prevention of infections to the healthcare workers, of course, and MDROs transmission during orientation, for example, or periodic educational updates for healthcare personnel. Make sure that all healthcare personnel in your institution are up to date with their knowledge with regards infections, multi-drug resistance, reporting, emerging and re-emerging diseases or pathogens. Number five and last, monitor antimicrobial susceptibility reports, because this will give you a huge, true reflection of how the treatment, how the usage of antibiotic is impacting on your institution, and how does the whole process of patient service is coming through from the nursing service, from the doctor's decision about the doses and duration of the antibiotic, and from the clinical pharmacist intervention to these reports from the lab releasing their final reports on time, so we can get an early intervention to prevent infections. Thank you so much, guys. I believe I'm done from this lecture. Any questions? Oh my God, people also need to. Yes, MRSA is a resistant bacteria, يا ريهام. Any questions? شكرا انتو. Thank you. Thank you guys. شكرا، كويس إنها ممتعة. Thank you so much. الله يعافيكم. حاخذ بريك شوية أطول، نرتاح، نجي 11 و20 دقيقة. الله يعافيكم. Thank you يا جماعة. Thank you for the nice words. Thanks. طب good، الحمد لله. Thank you so much. الله يسعدكم. I'll see you in 20 minutes. So نخلص إن شاء الله على 12 وبعدها عندكم break من 12 ل1 زي دايما. Hopefully you enjoyed it يا جماعة. أهم شيء إنكم استفدتوا واستمتعتوا. الله يسعدكم. I'll see you in 20 minutes. أهلا. هلا فيكم، يعطيكم العافية. صوت واضح ولا واطي؟ لا. تمام تمام. Thank you. شكرا. أوكي. طيب يا جماعة، يعطيكم العافية جميعا. Thanks for joining again on time. And في سؤال هنا: how we treat and manage ال MRSA تبع المحاضرة اللي قبل. ال MRSA لها decolonization. It has a decolonization. Let's say مو a procedure, decolonization protocol إن صح التعبير, if I could say. The MRSA transferred with contact, direct contact. So you have to adhere properly to contact precautions and environmental hygiene, hand hygiene, good selection of antimicrobial therapy could help. And of course, based on the patient age, patient clinical status, you could go through an MRSA decolonization protocol or not, because some patients cannot go through that protocol and some they can. So based on that, you can decide if you could go with this protocol or no. But yes, MRSA patient could be controlled, but they need strict implementation of infection control principles and measures. التريتمنت للMRSA يا ريهام is decided by a treating physician, not by an infection preventionist. So as an infection prevention, we cannot say what kind of antibiotic that the patient should go through. However, the treating physician can do, and the infectious diseases consultant with comparison collaboratively with the clinical pharmacist as well. طيب أنا جاوبت. I have answered كل الأشياء اللي تتجاوب, all the questions that could be answered. Thanks for joining again. And this lecture is like light one, خفيفة ما هي ثقيلة, light lecture. It is basically based on a public health point of view. So اليوم إحنا ما شاء الله يعني we spoke a lot about microbiology and public health and بكرة أو بعد بكرة immunity. So it's not really infection control, but it's all the things that you should know as an infection preventionist. So today, last lecture with me today is the microorganisms that required immediate review, management, control, let's say detection, let's say teamwork, if I could say, this kind of interventions is necessary in this part. The outline today is a short introduction, actually, to the topic and the public health role in such a case of microorganism reporting procedure, reporting protocol, notification policy, if I could say, all this kind that comes under the role of the public health and infection prevention. And Saudi Arabia, top 10 causes of death and reporting diseases that causes death, that cause morbidity, that cause mortality, actually is quite indicated to have a relation and association with the top 10 causes of death in Saudi Arabia. Communicable diseases, outbreak in healthcare facilities, as a short brief, outbreak management and facility, because I believe that most microorganisms, reportable ones, are harmful and high risk, labeled as a high risk pathogens, and a single or two of them, cases of them, could be an outbreak in the facility. So therefore, we have to come through outbreak size categories. We have to discuss some of common notifiable diseases, types of notification process to MOH whenever is necessary, and finally, the detection and control measures whenever indicated, whenever needed to be implemented. So as an introduction, the epidemiology and incidence of specific microorganisms can vary widely depending on the environment, depending on the commensal flora, and the transmission risks. So epidemiology incidence is differ based on many factors. Sometimes it's related to specific microorganism. Sometimes it's related to an environment, specific environment. Sometimes it's related to a specific host at risk, for example. So organisms that may be common in one geographic area may be uncommon in another. هذا الفكرة. However, given this ease of international travel, few microorganisms are confined to specific countries or areas, and the possibility of the regional disease to be escalating into a pandemic threat has been well documented, not only for influenza, for example, زمان مثل الانفلونزا H1N1 مثلا, but for other infectious diseases as well. And here we can never forget the pandemic during COVID-19 and how was this, the situation was really disaster in the whole world. So from this, traveling issue could be a risk factor in this case. We are talking about microorganism notifiable ones based on geographic areas, based on specific countries, regional diseases, escalating sometimes from place to another. Fortunately, today laboratory techniques are similar in the whole world, and most parts of the world and technology makes information shared possible in most areas. So whatever comes popular in Africa, we can know it easily. Whatever can be common in the US, we can here in the Middle East know it and be prepared and communicate and share. We can share also our strategies, our plans, our thoughts, how we mitigate and how we can come up with a better implementation, better preparedness, better readiness for such a kind of risks, if I could say. Additionally, international genesis, such as agencies such as CDC and the World Health Organization, WHO, assist with laboratory analysis of unusual or epidemiological important pathogens. So CDC, WHO are aiding everyone in the infection control field to have the data, the international data that we could share, we could learn from, and we could implement as well in our region. Clinical microbiology laboratories must work closely with the clinician when unusual pathogens are detected and pandemic threats identified, or during an outbreak investigation period, or during environmental sampling collection, for example, or time investigation. So clinical microbiology laboratories must work closely with the clinicians, with the infection control team, and report as soon as possible whenever they indicate unusual pathogen and detect a pandemic threat identified. The primary objective I can't read that from the 1 minute. Okay. Outbreak, I guess, management is the primary object in outbreak management is to first protect public health by identifying the source and implementing the control measures to prevent further spread. So if we are talking about microorganism that is notifiable or has the alert to be notifiable or مثلا detect or raised or prevent, we have to say like, okay, it's an outbreak committee need to be set it, and this outbreak committee objectives are to protect the public health by identifying those sources and implement control measures to prevent further spread of this threatened organism. Number 2, regional command and control center اللي هو CCC. Commander is the outbreak management team leader, okay, in this case, and shall order the formation of outbreak management team, OMT, upon variation of the outbreak, meaning since the outbreak has been verified, confirmed, the RCCC should be the chairperson for the command, should be the person or the leader for this investigation. Central CCC provides logistic and technical support as needed, so they are really helpful source. They can help infection control department in many aspects. They can support even with the meningeal level type, like they can give you the authority to manage your things, to order extra personal protective equipment, to support your vision within your hospital management leadership team, etc. The outbreak management team to be formed from the RCC and the infection control department or unit at the healthcare facility for primary control of the outbreak within the facility. So these are the objectives of an outbreak management team initiation. The OMT has the authority also for all restrictive measures to be contained the outbreak, including hospital units, closure even, if that could be taken Total closure of the healthcare facility in an outbreak must be coordinated and approved by the Central CCC. Central CCC outbreak management team may be deployed to assist in a medium or a large outbreaks cases. Okay, whereas small outbreaks could be handled and managed by the hospital itself, infection control team. Upon request from regional or central CCC, field epidemiology training program shall conduct outbreak investigation to provide timely information, because the time here is crucial. In outbreak, the time is very, very crucial. And confidence of information, of course. Variation of the information, of course, is quite crucial as well. And guide that assist in the control of the outbreak. All healthcare facilities within the region to be timely informed and of the outbreak, and necessary preventive measures are implemented. The preventive measures actually should be implemented immediately in extra level, to avoid any further spread and to avoid worsening scenarios. Administrative support at the facility, health sector, regions, and ministerial level has to be assured as well. So what is the public health role? What is the public health role in the reporting, notifying microorganisms that is really, really harmful? So public health is defined as the practice of preventing disease and promoting good health within a group of people, from small communities to entire countries. This is what public health defined as. Public health activities and services. The common themes describing public health encompass organized efforts to first protect and promote human and environmental health. These are the activities and the services. Prevent communicable and chronic diseases. And number 3, improve health and access to health services for persons with limited resources. So in this case, we understand the public health role, activities, services within infection control perspective. Public health services include vaccination, for example, programs, human immunodeficiency virus, acquired immunodeficiency virus, مثلا ال HIV والAIDS, counseling and testing, for example, public health community education as well, outbreak investigation and case follow-up too, tuberculosis and sexually transmitted infection treatments. They involve in all these aspects or services. And more recently, biotourism, for example, emergency levels and pandemic preparedness, leadership and response. So from these services, you can understand the role of public health in notifying disease or re-emerging microorganism, or reporting a high-risk pathogen in a facility to the higher level. Public health role also classified the importance of the disease based on different aspects. First, based on the mortality that this disease could cause, and based on the morbidity of this disease could cause, and based on the cost and socio-economic impact of this disease which caused on the society. And finally, based on the preventability of this disease. From these aspects, the public health could classify the importance of the disease from one to another. Reportable or notifiable diseases are those of public interest by reasons of their contagiousness, severity, and frequency. So whenever we have an organism that is considered to be contagious, cause severe infection, or frequently appeared in a place or a local place or geographical area, this means, okay, this is an alert, this is a red flag, this is something that we should consider. Moving to the Saudi Arabia top 10 cases of disease, and we care because we all تقريبا work here, and if we get a better view about how important is infection control, public health, and microbiology, but that, I think you can shift your mind and look at the thing in a different way after this informations, because the top 10 cases of death in Saudi after the latest statistics from Wiqaya, the CDC, Saudi CDC, first is a heart disease, as we know. Second is injuries. Third is a stroke. Fourth is the chronic kidney diseases. Number 5 is lower respiratory infections. Number 6, Alzheimer's disease. Number 7, conflict and terrors. Number 8, cirrhosis اللي هو تليف الكبد. Number 9, neonate disorder. Number 10, diabetes. And as infection preventionists, we can find the risk factor here, number 5, lower respiratory infections. And most of the time, the lower respiratory infections had many causes, many reasons, but I'm not going to mention them all, but most of them is cross-infection with droplets or airborne diseases. For example, also antibiotic lung therapy, for example, using non-sterile respiratory equipment, non-clean, non-decontaminant sterile equipment. So from this slide, you can understand that infection prevention is crucial. And this is considered to be in the middle of that. It's a top 5. We're talking about top 5 causes of diseases in Saudi Arabia, which is preventable, which can be deleted by prevention. It's not like other diseases. If you got my point, this is really preventable and we can make it true. So it's hard to understand like, oh, wow, low respiratory infections is number 5 cause of disease in Saudi Arabia. And it is something preventable. So we could make it. So lower respiratory infections usually comes from the communicable diseases. And communicable diseases, they causes an outbreak in healthcare facilities. So speaking about those things, let's go through disease outbreaks and how can infection prevention prevent low respiratory infections, the cause of disease, the cause of death, number 5 in Saudi Arabia. So it is the disease occurs occurrence in a population above the normally expected rates and any given time or location. The expected number of cases can be determined through ongoing disease surveillance. So surveillance is a tool, is a quite helpful tool to guide us if there is any thing goes wrong, if there's anything is increasing, any growth is increasing, any harmful pathogen is presenting. So surveillance is a quite important tool. This involves the systemic collection of numerator and denominator data using standardized case definition and surveillance methods, of course. Additional diseases may become reportable as a result of current outbreak. For example, influenza cases and newly emerging conditions such as SARS, MDROs, MRSA, VRE. Those are additional reportable diseases. Whenever you feel like, oh, the result is coming high, the lab report is having too much positive specimen, then yes, we can say it's newly emerging conditions and it's an outbreak. Outbreak size categories depends on, number 1, limited to a single unit in a single healthcare facility. So there is a there is a size of the outbreaks. Number two is multiple units in a single healthcare facility. And we are going bigger from A to D. Okay. Letter C, multiple hospitals in a single city. And the biggest thing is multiple hospitals in a multiple city. Common notifiable diseases. What are the common notifiable diseases? Now, regulations related to the reporting of the notifiable diseases exist to protect the general public from health threats, of course. And infection prevention plays a vital role in the notifiable diseases reporting system, of course, often handling the reporting for their organization. For example, MERS-CoV, severe acute respiratory syndrome, SARS, monkeypox, and Ebola. Most probably, those are the high reportable diseases. Pulmonary TB, HIV, Clostridium difficile as a public health disease, surveillance and reporting, they are important. For the surveillance purposes, for the reporting, so the public health could know the situation of the community with the number that they have of TB positive cases, HIV positive cases, Clostridium difficile cases, so they can track them, monitor their presence in the community, trying to give them the best treatment so they can get rid of these diseases. Notification to the decision maker in MOH in case of meningococcal meningitis and cholera, plague, yellow fever, and poliomyelitis disease. Types of notification process to MOH. What are the types? There are 4 types or 5. Let's check. 4 types. Yeah, 4 types for notification process to the MOH according to the المديرية العامة لمكافحة العدوى. First, immediate. So when you have to report or notify the MOH immediately, this is for class 1 disease that needs to be immediately immediate action. Notification is for fax or phone or electronically through حصن. Okay, the Health Electronic Surveillance Network is an electronic reporting system was developed for reporting infectious diseases. When the case is reporting in Hissa, the Public Health Department in the region and the Ministry of Health headquarters can view it immediately to take the appropriate action. Second type of notification is weekly, and this is for class 2 diseases, which will be sent from the health units to the regional health affairs. Number 3, monthly reporting or notification process. This includes all infectious diseases notified to the regional health affairs, which in turn notify the Deputy Minister for Public Health. Number 4, weekly zero reporting. And this one is from all acute flaccid paralysis cases in children less than 15 years, all suspected measles, rubella, and mumps cases, H1N1 cases. Notification are sent from regions and other sectors to infectious diseases directorate. Now, after knowing and understanding the notification process and type of outbreaks, sizes of the outbreaks, what is the role of public health, and how it's really, really important. And when sometimes we decide, oh, this is an outbreak, this is not, this is a notifiable thing, and this is not, based on the data we have showed. Now moving to the detection and controlling measure in case we have an outbreak, in case we have a notifiable diseases. So first, we can take MERS-CoV as an example. For example, in this case, let's say we have a MERS-CoV case. So all measures should be in place to prevent the MERS-CoV outbreaks in healthcare facilities per the published national guidelines. So there is a MERS-CoV guidelines from the MOH. This should be followed. This policy should be implemented whenever you have a case of a MERS-CoV, for example. Number 2, risk assessment. Risk assessment should be performed early by the outbreak management team once secondary transmission have been confirmed in the healthcare facility. So the risk assessments should be started. Number 3, outbreak management team to meet at least daily so they can discuss all the outbreak management issues, obstacles, and how can they make this could be better and better. Number 4, the rapid response team, RRT, will carry out its actions of inspection, on job training of staff, distribution of training materials as well, and immediate corrections of infection control practices. Number 5, all persons who had contact with a confirmed case in the healthcare facility and community to be identified and traced. The infection control unit of the facility will identify and trace all healthcare workers who had exposure to a patient diagnosed with a MERS-CoV and will assess their health daily for at least or a total of 14 days. And the Department of Public Health and the Regional Health Directorate will trace all household of the other community contact of the patient with a confirmed MERS-CoV infection for the total of 14 days as well, and testing for MERS-CoV in symptomatic individuals only. Line listing recording for household and healthcare contacts will be updated daily. This is really important, helping us to trace, control the community that is under risk. Okay, and healthcare providers on the contact list should not report to work and inform their superiors if they have symptoms suggestives of MERS-CoV infection, should sign clearance in a logbook at a triage area when reporting to work immediately. All healthcare workers' contacts, low and high risk considered contacts, should be tested for MERS by PCR not less than 24 hours after exposure. Only those with high risk exposure may be tested again within 3 days of the previous negative test. Any symptomatic healthcare worker on the contact list should be tested too, because he is asymptomatic, he or she. Repeated testing of symptomatic and MRSA negative healthcare worker is at the discretion of the outbreak management team, TIMIT. And healthcare workers or household of the contact list should not travel outside the kingdom and do not attend mass gathering events like Hajj or Umrah, for example, for 14 days from the time of exposure. Movement within the kingdom should be notified to the outbreak management team before that. Number 6, outbreak management team will put in place the control measures, including proper patient placement and compliance with infection control measures. The infection prevention and control audit team will conduct audits on the healthcare facilities within the city to ensure preparedness is in place. Number 8, emerging community clusters to be managed by the regional CCC. Home isolation should be considered for stable patients, and those that require hospital care should be admitted to a MERS designated center. These are the references for this topic, and we have done. Thank you, guys. Any questions? Okay, good. العفو. Thank you, guys. So I think we will go. عفواً، ألف عفو. شكراً جزيلاً. We can go for a break from now to 1 o'clock. And next.
الصوت والشاشة واضحين؟ طيب الصوت واضح كويس؟ ممتاز. Excellent. If you can see and hear me, excellent. Welcome back and good afternoon. My name is Sumaya Jul. I'm infection control specialist. My time with you today will be from 1 to 3:30, so we will finish our lectures today by 3:30. I'm an infection control practitioner at Dr. Sulaiman Al Habib Hospital here in Riyadh. And today I will discuss with you a subject about the microorganisms and how infectious disease can be contributed by these microorganisms. And I will try my best to translate all lectures in Arabic. And there is a time for your questions will be at our by the end of our lectures or in between, just to make things clear. مساء الخير على الجميع. معكم سمية جلون، أخصائي مكافحة عدوى. إن شاء الله محاضرتي اليوم حتكون خفيفة عليكم إن شاء الله بإذن الله، من 1 إلى 3:30. فحنخلص كل محاضرات اليوم الساعة 3:30، وحيكون في مجال للرد على الأسئلة غالبا نهاية المحاضرة اليوم، أو مثلا إذا في أجزاء غير واضحة بالنسبة لكم. أعمل كممارس مكافحة عدوى في مستشفى دكتور سليمان الحبيب بالرياض، وأحمل شهادة ماجستير في مكافحة العدوى جامعة الفيصل. محاضرات اليوم حتكون عبارة عن الكائنات الحية الدقيقة في سياق تفعيلها لموضوع أو المفهوم ومبدأ العدوى، وكيف تكون هي السبب في أغلب الأمراض المعدية وحتى الأمراض العادية، عبارة عن هي تراكمات أو استعمار لأنواع مختلفة من البكتيريا والفطريات والفيروسات. إن شاء الله بإذن الله أحاول قد ما أقدر أتكلم أو أشرح في المحاضرة بالعربي بالإنجليزي ويكون الشرح واضح للجميع. So our lectures today will start with some objectives. The first one, characteristics of biology of bacteria, viruses, fungi, parasites, and allergy. And the second object will be microorganisms in the context of nosocomial infections. And the last one will be how to transfer or transmit from one object to another, or from person to another, or one patient to another. لذلك أهداف أو مواضيع محاضرتنا اليوم حتكون الخصائص الحيوية. اعتبروه درس علوم اليوم عن مكونات أو تفصيل مكونات البكتيريا، الفيروسات، الطحالب والفطريات والطفيليات. وأيضاً حنتكلم شوية عن الكائنات الدقيقة في سياق الأمراض المعدية، واللي غالباً تكون داخل المستشفيات أو تحدث داخل المستشفيات. وأيضاً حنتكلم على طريقة انتقال الكائن الحي الدقيق داخل المستشفى لإحداث العدوى. First of all, حنتكلم we will discuss about some characteristics of the biology of these microorganisms. At the beginning, we have to know that the study of microorganisms, which are too small to be identified by the unaided human eye with the naked eye, called microbiology. So some students of microbiology or some of you who studied microbiology will understand clearly what is the lecture about. And there are a number of types of different microorganisms. For example, bacteria, viruses, fungi, parasites, and algae. And as we can see in the picture, they are in different shapes, different structure, different components, different variants of doing the infection or causing the infections. And under microscope, they are really very different. These various organisms affect human body internally and externally, and also affect the environment. There are very few places in the world where microbes or microorganisms are absent. And some microbes or microorganisms can survive even in the harshest of the environments. So as there are creatures in our planets, they have some structure and they have components. They have to find the correct or the right environment to live in. And they have to create a good environment for them to live and to continue to rest, to eat, to discharge, and to reproduce, and to live a normal life until they are dead. So as like any other creature, they have a life cycle. علم الأحياء الدقيقة هو عبارة عن دراسة الكائنات الحية الدقيقة الصغيرة جدا التي لا تشاهد بالعين المجردة. طبعاً زي ما نشوف في الصورة أشكالها، أنواعها، هيكلتها ومكوناتها وتركيبها كله يختلف عن الآخر، سواء كانت بكتيريا أو فيروسات، طفيليات أو فطريات أو طحالب. معظم الكائنات الدقيقة الدخيلة على جسم الإنسان تسبب عدوى أو اختلالات أو اعتلالات في الصحة، سواء كانت داخل الجسم أو على سطح البشرة خارجياً. وأيضاً تؤثر في الأماكن، يعني تؤثر في الجماد، تؤثر في البيئة، تؤثر على النبات، تؤثر على الجماد، تؤثر على البيئة التي تكون حول الإنسان. وهناك يعني أماكن قليلة جدا جدا اللي يندر فيها وجود الكائنات الدقيقة، سواء كانت بكتيريا أو فيروسات أو طحالب وفطريات أو طفيليات وإلى آخره. يعني غالبا مكونات كل الأشياء في العالم حولنا هي عبارة عن كائنات حية دقيقة، بغض النظر عن إنها كانت ضارة أو نافعة. إذا جينا نتكلم عن البكتيريا ككائن خلينا نقول يمثل، خلينا نقول 75 تقريبا في المية من مكونات هذه الكرة الأرضية. البكتيريا هي عبارة عن خلية وحيدة واحدة، لكن هي عبارة عن مجموعة من الخلايا الصغيرة جدا اللي تدخل ضمن مكونات واحدة خلية. يعني هي عبارة عن نقاط صغيرة جدا عشان لما تتجمع مع بعض نقدر نقول عليها هذا تركيب الخلية اللي هي الخلية البكتيرية. موجودة في كل مكان حولنا وفي كل شيء من المكونات حولنا، موجودة في أعمق نقطة في البحر وأعلى نقطة موجود فيها مثلا أعلى الجبال، أو أعلى مثلا المناطق في أو الجغرافية على سطح الكرة الأرضية. من مكونات الخلايا البكتيرية إنه هي ما عندها غلاف خارجي، والمكونات الداخلية حقتها كما نشاهد في الصورة متعددة عديدة، لكل مكون داخلي وظيفة، وبرضو خلايا جوا مكوناته تساعده على إنه يؤدي هذه الوظيفة. وفي عندي قسمين من الخلايا البكتيرية تسمى إيوكاريوتيك وبروكاريوتيك، هذه اللي تكون فيها يعني خلية عندها ممبرين اللي هو غلاف، والثانية بدون غلاف. طبعا كل واحدة لها وظائف، لها فائدة، لها نفع ولها ضرر ولها مميزات ولها مواصفات تختلف عن الأخرى. وطبعا ما نقدر ندخل لشرح التفصيل الدقيق لأنه يعني مو أغلبنا يقدر يعني أو يستوعب أو مثلا يكون جزء من عمله تفصيل تكوين الخلية البكتيرية. لكن حنشرح بشكل عام إنه البكتيريا اللي تكون عندها جدران أو غلاف هي طبعا مختلفة تختلف تكوينها من نوع لنوع، من فصيلة لفصيلة، وتختلف في أدائها لطريقة الوظيفة اللي يعني متخصصة فيها. وكيف تقدر يعني تنتقل من كائن لكائن، كيف تقدر تسبب العدوى، كيف تقدر تسبب النفع في هذا المكان أو وجودها جزء من تكوين هذا العضو وإلى آخره. فيعني باختصار ما أبغى أدخل كمان في المصطلحات العلمية اللي تكون جزء من مكونات الخلية البكتيرية لأنها ما أتوقع أنها هي صعبة عليك سهلة عليكم، لكن أشهر جزء ومكون معروف دائما أو يدرس حتى في المناهج التعليمية للتخصصات العلمية أو مثلا مادة العلوم بشكل عام في المدارس، جزء اسمه ميتوكوندريا اللي هي موجودة في الصورة على اليسار. الميتوكوندريا هي عبارة عن جزء أساسي من مكونات كل خلايا البكتيريا، تحتوي على جزء بلازمي أو غطاء بلازمي، تحتوي على جدار الخلية، وبعدين تحتوي على كابسول اللي هو الكبسولة اللي تكون شكل هذه الميتوكوندريا، تحتوي على أهداب تساعدها على الحركة، تحتوي على صوت اللي لونه أحمر هذا اللي في الأسفل يساعدها برضو على الانتقال من مكان لمكان. فيها مكونات مثلا تساعدها على الاستمرارية في الحياة، إنها ما تموت بسرعة وإلى آخره. فجزء من مكونات الخلايا البكتيرية اللي هو الأهم اسمه ميتوكوندريا. لو تبحثوا عنه حتلاقوا أشكال وألوان من يعني الكائنات الحية الدقيقة اللي يكون موجود داخلها ميتوكوندريا، وهو جزء بسيط جدا من تكوين الخلية اللي تخيل يعني عظمة الخالق في خلقها داخل الخلية البكتيرية دي أنا حتى ما أشوفها بالعين المجردة من كثر ما هي دقيقة وصغيرة. So if we are talking about the bacteria which build or contribute to create most of all of the things around us, we should say about it that they are a group of microscopic single-celled organisms that live in enormous numbers in almost everything in the environment, from the earth, from the deep sea vents to deep below earth or high mountains, and also in the human body, in the plants, in all of the industrial chemicals or products or even food or water places in our planet. So a few places in the earth that doesn't contain any type of bacteria. If we want to differentiate between types of bacterial cells in the shape, we have to know that there are two types, eukaryotic and prokaryotic. One of them has membrane, cell membrane, and the other one doesn't have cell membrane. Bacterial cells are prokaryotic cells and are different from animal cells in several ways. One central difference is that bacterial cells lack of intracellular organelles like a nucleic and mitochondria, which are present in animal cells. يعني في بعض الكائنات البكتيرية أو الخلايا البكتيرية ما يكون عندها الوضوح للميتوكوندريا، لكن الفرق بين الخلايا البكتيرية كهيكلة اللي هي خلايا بكتيرية عندها جدار خلية أو غلاف، والأخرى لا تحتوي على غلاف. In order to examine bacteria in detail microscopically, they must undergo a process called gram staining عشان أفرق بين أنواع البكتيريا. أو مثلا عندي مرض معدي وصل لي كنتيجة من المختبر أو حالة عرضية انتقال لعدوى. عشان أفرق بين إنه هذه البكتيريا إيش نوعها، كيف شكلها، أعرف مين هي، أتعرف عليها، في المعامل خطوة مهمة جدا لاكتشاف أو لتحليل أي عينة موجودة عندنا، اللي هي في البداية أول شيء جرام ستينينج، اللي هي صبغة جرام. هذه الصبغة لما أجي أحطها على الشريحة أو العينة اللي تكون تحت الاختبار توضح لي أول شيء لون الصبغة. إذا طلعت لون الصبغة حمراء حقول نيجاتيف جرام ستينينج، أو إذا طلعت لونها بنفسجي حيكون بوزيتيف جرام ستينينج. طبعا ولكل واحدة في موظفينا مختبر حينبسطوا على هذه المحاضرة إنه كل واحدة لها مزايا، جرام ستينينج بوزيتيف جرام أو نيجاتيف جرام. وعلى هذا الأساس حعرف إنه تحت الميكروسكوب في أشكال مرة جدا مختلفة للبكتيريا، ممكن تكون دوائر كروية أو مثلا عصوية أو شكلها فايبرو اللي هو الشكل الثالث في الصورة، أو كوكوباسيلس يعني بيضاوية، أو سبيريليوم اللي هو زي الدودة أو متموجة، وفي اللي هي آخر واحدة السبيروشيت. وطبعا بناء على ذلك كل واحدة في لها مواصفات وتكوين داخلي هيكلي وأيضا وظائف حيوية، ولها دور في إنها مثلا إما تكون نافعة أو ضارة من ناحية تأثيرها على الوجود في المكان اللي تكون فيه، سواء الجماد أو النبات أو الإنسان أو الحيوان. أيضا من المميزات اللي تكون اللي نعرف فيها الخلايا البكتيريا تحت المجهر لما تكون جرام بوزيتيف أو جرام نيجاتيف، إنه في واحدة منهم يكون سيل وول ستراكشر، هذا ما هو موجود. بالإضافة إلى إنه the choice of antibiotic therapy will be based on the degree on whether the organism is gram positive or gram negative. فهذه يعني خلينا نقول عليها نقطة متقدمة في موضوع اللي هو تفصيل مكونات الكائنات الدقيقة، إنه حتى العلاج لما أجي ألاقي عندي حالة عدوى موجودة لمريض منوم عندي، بناء على نتيجة التحليل اللي توصلني من المختبر أعرف إيش نوع البكتيريا وإيش اسمها، أو مثلا إيش خصائصها وإيش نوع العلاج أو المضاد الكيميائي، المضاد الحيوي اللي استخدمه لعلاج هذه الحالة. لذلك تم تقنين صرف المضادات الحيوية وخاصة اللي ما تكون موصوفة بالطريقة الصحيحة للمريض الصحيح وفي الوقت الصحيح والكمية الصحيحة. فهذا برضو يعني تكوين هذا فهم تفصيل تركيبة الكائن المعدي اللي هو خاصة البكتيريا لها دور في إني أنا أعرف أصرف أي نوع من المضادات الحيوية. لماذا؟ يعني هي شوف الشريحة باينة قدامكم إن شاء الله، بس يكون الصوت واضح. إنه جاني اتصال. طيب واضح. خلينا نقول خليني أسألكم لماذا؟ معلق عندي شوي. إحنا هنا اللي عنده خلفية أو يعرف يقول لنا لماذا. أنا آسفة بس لازم أفتح الشات عشان أشوف الإجابات. أوكي. اللي عنده خلفية أو درس مثلا معانا دكاترة صيادلة، أيوة السؤال يقول لماذا صرف العلاج أو المضاد الحيوي بالذات، صرف المضاد الحيوي يكون بناء على نوع أو نتيجة أيوة النتيجة اللي تجيني من المختبر اللي توضح لي فيها نوع البكتيريا. اللي هو الجزء الأعلى من الكائن، أو هو المكان اللي يكون فيه زي الحساسات، يعرف فيه هذا الفيروس فين المكان اللي استقر فيه في جسم المريض عشان أبدأ أعيش في هذه البيئة. مثال some important examples of viruses and the diseases they cause. مثلا DNA viruses، الفيروسات اللي عندها double nucleic acid تسبب لي مثلا smallpox, chickenpox, sore throat, conjunctivitis or serum hepatitis أو warts. الفيروسات اللي تكون تحمل ال RNA genome تسبب لي مثلا influenza, measles, mumps, rabies, AIDS, meningitis, Lassa fever, also yellow fever, encephalitis وإلى آخره. فالأساس اللي إحنا كلنا لازم نعرفه viruses are not part of the component of any normal subject or a human or an animal or a plant, but it's transit. It will come, it will do the damage and leave. Viruses are classified based on their size and shape. Also, if they are having envelope or no. In enveloped viruses such as influenza or hepatitis C viruses, the nucleocapsid is surrounded by a lipid bilayer from the modified cell host cell membrane and studded with an outer layer of virus envelope glycoproteins. Non-enveloped viruses lack of this membrane. So they have a capsid and they have also cell membrane called envelope to make sure that they are in double protection. So they will do the damage at a higher level more than the bacteria cells. In many cases, DNA viruses use cellular enzymes for synthesis of their DNA genome and mRNAs, messenger RNA. All viruses utilize standard cellular ribosomes and translation factors for synthesis of their proteins. في هذا الباراجراف مكونات يتكلم عن ال DNA وال RNA، مكونات المكون أو الهيكل الخاص بكل فيروس، كيف هو يقدر يشتغل على باستخدام الأنزيمات اللي هو يشتغل على الخلية اللي تقول يلا هنا هذا المكان اللي أنا أقدر أعيش فيه بغض النظر عن أنه أنا سأحدث الضرر أو لا، سواء كان في الإنسان أو الحيوان أو النبات. Viruses has evolved to become extremely effective at nucleic acid delivery to exact cell types, while evading immune surveillance by an infected host. This ability makes them desirable for engineering viruses vector systems for the delivery of the therapeutic genes. Several types of viruses, including retrovirus, adenovirus, and adeno-associated virus, herpes simplex virus, has been modified in the laboratory for the use of gene therapy applications. يعني كمان بيقول لك إنه الفيروسات لما تكون تحت مثلا إشراف استخدام علمي لنطاق معين، ممكن أستخدمها في بعض العلاجات اللي تساعد في تحسين حالة المريض، ولكن اللي هي في صورة أمصال، اللي هي تكون مثلا سيرم، أو لما أحطها في التطعيم، لما أخذ عينة من الفيروس وأحطه في تطعيم، لما أحط مثلا كائن أو مركب كيميائي مع نوع من الفيروس أو جزء من سلالة الفيروس في تركيب مثلا مكون تجميلي وإلى آخره. لكن بشكل عام الفيروسات هي عبارة عن كائنات دقيقة دخيلة على كل مكان هي تزوره، فهي ما راح تكون نافعة في أغلب الأحيان، لكن لما تكون تحت إشراف معين بدراسات معينة وتجارب وتطبيقات وإلى آخره، ممكن تدخل في اللي هو العلاج الجيني أو تطبيقات العلاج الجيني اللي يكون فيها مثلا بعض أنواع علاجات السرطان عافانا الله وإياكم، أو الأوتو إميون ديزيز وإلى آخره، مثلا كما الجينيتيك ديس أوردر اللي هو الاختلالات الجينية في بعض الحالات الخاصة، يعني مو في كل الأماكن أو مو في كل الأماكن. إذا انتقلنا إلى الفطريات، هي عكس تقريبا البكتيريا. الفنجاي are eukaryotic cells and have complex internal structure، يعني برضو فيها مكونات شوية مختلفة عن البكتيريا. Fungal walls are made of a matrix of components that are embedded and linked to scaffold of fibrous, load-bearing polysaccharides, and almost all of the chief cells wall components of fungal pathogens are unique to fungi and are not present in human or animals or mammals or plants. As the result, the immune systems of animals and plants have evolved to identify many of the conserved elements of single walls or fungal walls. Also, fungal species are approximately more than 300 types, and also all of them are harmful to humans. For example, or for the main famous fungi species that are very dangerous for each or for any person or any human is Candida species, either Candida aureus or any other type of Candida. الكانديدا لو كانت موجودة في عينة من عينات الدم، هذا البيشنت يعامل معاملة خاصة جدا، لأنه بسرعة لازم نلحق قبل ما يروح فيها. Candida species is the commensal of the skin and the gastrointestinal or gastrointestinal tracts of humans, and can also cause vulvogenitis, respiratory infection in the immunocompromised, oral thrush and sepsis. sepsis هي معناته إنه في كانديدا في عينة الدم ولازم نلحق المريض. خلاص تخثر في الدم، فحالته حتكون من سيء لأسوأ. مع إنه سبحان الله في بعض الحالات أو مو في بعض الحالات جزء من يعني العجب في هذا الكائن حق الكانديدا إنه هو جزء طبيعي من مكونات المهبل. إذا لقينا عينة مهبل أو عينة من سوائل المهبل فيها فطريات اسمها كانديدا، هذه تعتبر بيئة طبيعية لتواجدها، إلا إذا كانت متواجدة بعدد كبير وفيها يعني تفاصيل أخرى. لكن بشكل عام هذا النوع الوحيد من الفطريات اللي قد يؤدي إلى الوفاة، خطر جدا على حياة الإنسان. لكن وبشكل عام الفطريات يعني وجودها في جسم الإنسان حتى أيضا في بعض أنواع النباتات هو ليس جزء من مكوناتها زي بعض البكتيريا، فهي تجي تحدث الضرر ويمكن علاجها ويمكن أيضا تفادي حدوثها في حالات الوقاية طبعا. ومكوناتها مستخدمة كثيرا في الاتجاه اللي هو الرعاية التجميلية أو المواد التجميلية والأدوات التجميل، وأيضا العلاجات لبعض الحالات الصحية الجلدية، كثير فيها كائنات فطرية. ولكن الأخطر بشكل عام سلالة الكانديدا. طبعا في أكثر من 300 نوع من الفطريات، واللي تكون ما هي يعني وجودها غير مناسب لصحة الإنسان، يعني خطر شوية عليها. also كمان في كانديدا أوريوس. هذه الكانديدا أوريوس كمان أشد وأخطر. emerging fungus that presents a grave global health threat. And healthcare facilities in several countries have reported that Candida aureus has caused severe illness in hospitalized patients. Candida aureus is often resistant to multiple antifungal drugs. يعني مقاومة لبعض المضادات الفطرية اللي هي إيش؟ الكانديدا أوريوس. فبناء على ذلك ما أقدر أوصف علاج في مضاد فطري لكانديدا أوريوس واحد محدد، لا، لازم يكون مضاد فطري معين عشان يقدر يقضيها على هذا النوع من العدوى. Fungal isolates from clinical samples can broadly be split into either yeast or mold depending on their predominant growth form. يعني أيضا يعني اللي ما يعرف بشكل عام الفطريات تنقسم إلى ييست ومولد، كل واحدة لها خصائص، لها مكونات، لها هيكلة، لها ألوان، لها وظائف، وأيضا سلالات مختلفة تجي من تحتها بناء على نوع مثلا الضرر أو النفع اللي حتحدثه. وفي لها ظروف خاصة بيئية تعيش فيها. يعني في عندي demographic fungal مثلا تحتاج 25 درجة حرارة 25 درجة مئوية عشان تعيش، عشان تنمو فيها، بينما هناك أنواع أخرى من الفطريات تحتاج درجات حرارة أعلى عشان تكون البيئة المناسبة لتواجدها أو إنها تعيش وقت أطول. في عندي كائنات في عندي فطريات يكون شكلها كوكاي يعني دائرية. في كائنات أو في فطريات يكون شكلها نقول عليها هيستوبلازما، يعني شكلها إذا عصوية أو مثلا تكون لي زي كده دوائر مستعمرات ولكن ليست بشكل دائرة، يعني زي شكل الوردة تسوي لي هذه تحت المجهر ممكن أشوفها. في opportunistic fungi اللي هي تكون مثلا زي الكانديد albicans، تكون انتهازية، تتواجد أو تخلق وتتكاثر في بيئة تحتاج مثلا رئة مريض يكون فيها أنواع من البكتيريا معينة مع درجات حرارة، مع جفاف مثلا أو سوائل كثيرة، وأيضا في هذه الرئة يكون فيها مثلا بلغم كثير، فيكون إيش بيئة مناسبة جدا لخلق وبناء وعيشة هذا الكائن الفطري. فهي تحتاج مثلا خلينا نقول ظروف بيئية معينة تجرو لتنشأ، وكل جزء من مكوناتها وكل مثلا الظروف البيئية والشكل واللون والحجم والمقاس وكل شيء لها دور في إما إحداث الضرر أو اللي هي تنقلب إلى نفع. لاحقا إذا تكلمنا أيضا على الباراسايتس اللي هي الطفيليات. They are organisms that live on or in host organism and get its food from or at the expense of its host. يعني إما إنها تعيش في كائن مضيف تأخذ الغذاء منه، أو تعيش عليه وتنتقل وتروح تأخذ الغذاء كمان عن طريقه. وفي 3 أنواع منها three main classes of parasites are protozoa, helminthes وectoparasites. يعني هي أنواعها كده، يعني ما حيكون في طفيليات في نوع رابع مثلا. البروتوزوا microscopic one-celled organisms that can be free-living or parasitic in nature بشكل عام. Transmission of protozoa that live in a human's instinct to another human typically occurs through fecal oral route. يعني drinking unsanitary water or food. انتقالها واضح البروتوزوا. Protozoa that live in the blood or tissue of humans are transmitted to other humans by an arthropod vector. لازم يكون طائر كائن طفيلي طائر يطير، عشان كده ينتقل من مكان لمكان. For example, Plasmodium vivax through the bite of mosquito. تسبب لي إيش؟ الملاريا. هذا النوع الأول من الطفيليات، يعني هي يا إما إنها تكون مرة صغيرة ما أقدر أشوفها، أو إنها ممكن تكون طائرة تتحرك من مكان لمكان بالأجنحة. الثاني النوع من الفطريات اللي هي helminthes، أكبر شوية، multicellular organisms that are generally visible to the naked eye in their adult stages. وبرضه حنشوف صور لها شوية أكبر من اللي هي البروتوزوا. الإكتوباراسايتس اللي هو النوع الثالث can broadly include blood-sucking arthropods such as mosquito، يعني في أجزاء وأنواع منها تكون أقدر أشوفها بدون مجهر، أقدر أشوفها بالعين المجردة، ويكون مثلا أنواعها مشابهة للذباب، الناموس، البق اللي احنا نقول عليه نشوفه بعيننا، وخاصيته إنه يمتص الدم من جسم المريض وجسم الإنسان بشكل عام، وينقل فيه المرض من مريض إلى آخر عن طريق امتصاص الدم، يعني متخصص في القرص والدم. Arthropods are important in causing diseases in their own right, but are even more important as vectors. فهي ناقلة أكثر من مسببة يعني للمرض. On many different pathogens that in turn cause tremendous morbidity and mortality from the diseases they cause. مثال، هذه هي أنواع وأشكال الباراسايتس. الباراسايت ممكن يكون زي ما قلنا ذباب، حشرات طائرة، البق في الفراش، وفي منها اللي هي تكون الديدان الشريطية مثلا، اللي تكون موجودة في أنواع الأكل الغير مطبوخ جيدا. ممكن ديدان تنتقل عن طريق تراب النبات إلى الأيدي، إلى الملابس، إلى آخره. إيش عندنا أمراض ممكن تتسبب فيها الطفيليات؟ يلا خلينا نصحصحكم. أمراض تتسبب فيها الطفيليات، مثال إحنا قلنا ملاريا. إيش في كمان أمراض ممكن تنتقل بسبب الطفيليات؟ أمراض جلدية زي إيش؟ مو لازم تقول اسم الكائن أو الطفيلي، أعطينا أسماء الأمراض. طفح جلدي، سكين راش، أوكي، بس ممكن السكين راش يكون جزء منه مثلا إكزيما، فالإكزيما ما هي بسبب طفيليات. بالهارسيا إكسلنت، توكسوبلازموزيس إكسلنت، حمى الضنك إكسلنت، نزلات المعوية إكسلنت، سكيبيس، ليشمينيا. يرقان، إيش هو اليرقان بالإنجليزي يا سلمى؟ إنكلوستوما إكسلنت. الأمراض المعوية إكسلنت، جياريديا إكسلنت، جويندس الصفار، أوكي، مو الهيباتيتس، أيوة جويندس بشكل عام ممكن، بس لا مو مرة يعني زي التوكسوبلازموزيس والملاريا شيستوسيميا، ليشمينيا، شاغاز ديزيز، صح. داء الفيل ممكن، صح، اكسلنت. التهاب الكبد لا، مو عن طريق الطفيليات، الديدان الطفيلية، الديدان اللي هي الديدان بشكل عام يعني، ايوه. الاسكارس، الجايندس، اوكي. ملاريا قلنا، ملاريا. زيكا فيروس، نو. pinworm. انفلونزا الطيور لا، هذا ما هي بسبب الطفيليات، هذه بسبب فيروس الانفلونزا. يعني ما في طفيليات تسبب نقل لإنفلونزا الطيور. عرفت؟ فركز السؤال. حمى الضنك، يس اكسلنت، إجابة صحيحة. حمى الضنك، ملاريا. عندي مثلا هذه الكائنات اللي تكون في بيئة استوائية وتنقل المرض عن طريق فيكتور اللي هو ناقل، والبلهارسيا والشاغاز ديزيز هذه صح، كلها صح. يعني لازم يكون في حشرة سواء أنا أشوفها أو ما أشوفها تحت المجهر وتنقل لي المرض. هي بناء على ذلك أنا حاعرف نوع المرض أو الحالة المعدية. انتاميبا، سكيبيز أيوة ممكن صح. طاعون اكسلنت. ارتكاريا ممكن أيوة. إيش هو sleeping sickness؟ هذا لسه ما أعرفه أنا. أعرف حمى الضنك. إيش الحشرات اللي تسبب القرصات فتعطيني أمراض؟ ناموس. إيش يعطيني؟ ملاريا. إيش كمان؟ جرب. اكسلنت. اوكي. يلو فيبر، لسه فيبر. اقرأوا عنها كمان. ايوه انا كمان حقرأ عنها. اوكي ايوه الان عرفته. sleeping sickness. اوكي لانه شفته بالعربي كذا ضعت. دنجي فيبر ايوه حمى الضنك. اكسلنت. طيب فالان الغرض من السؤال اني انا افهم فكره ايش اللي نقله، فيروس ولا طفيلي ولا بكتيريا. فحعرف افرق بينها اذا انا عرفت مين السبب. السبب هو تلامس الايدي ولا حشرات ولا drinking unsanitary او contaminated food and water، ولا مثلا جاتني من مريض منوم في الغرفة اللي جنبي. I have to think logically about how the disease transferred to my patient or to me. لا قدر الله. Okay. Moving on to algae اللي هي الطحالب. الطحالب يعني ما ولا أحد يقدر يحصي ويعدها من كبر البحار والمحيطات. And they are very large and diverse group of autotrophic eukaryotic organisms, which range from uncellular to multicellular forms, and they are primary producers, which makes them a source of many nutrients and proteins. أنا من ناحيتي أشوف إنه الطحالب كائنات مسالمة جدا ونافعة بكمية مهولة أكثر من أي كائن موجود على سطح الأرض حتى الإنسان. والله إن الطحالب تنفع أكثر من الإنسان. They have many types of life cycles ranging in size from microscopic to micromonas species to giant kelps that reach 60 meters in length. On top of their ecological roles of oxygen producers and as a food base for virtually all aquatic life, algae are economically significant as a source of biocrude oil and as a source of food. بالإضافة إلى أنها مصدر للأكسجين وطعام للكائنات اللي موجودة في البحر، وعلاج أو يطلعوا منها ملايين منتجات التجميل وملايين منتجات الرعاية للجسد وعلاجات يعني مهولة. هي أيضاً مصدر للزيوت وأيضاً لترطيب البيئة بشكل عام. ليه؟ لأنها طبعاً تصدر أكسجين، كونها من النباتات اللي تعيش، كونها من الكائنات اللي تعيش داخل البحر وممكن تعيش على سطح الأرض بشكل عام. They have also a number of pharmaceutical and industrial applications for human. يعني مو بس تجميلي، لا في علاجات دوائية بمكونات الطحالب، وأيضا مكونات أو إنتاجات صناعية لها جزء أو جزء من تكوينها يكون الطحالب. فبشكل عام يعني هي ممكن تكون نسبيا بشكل ما وليس دقيق 75 أو 80% نافعة ومفيدة أكثر من كونها إنها تسبب بعض الأمراض أو الحالات العرضية المعدية. الأفضل طبعا من أنواعها اللي هي نقول عليها بلو جرين، الطحالب الخضراء المزرقة، for antibiotics and pharmacologically active components. من الأشياء اللي برضو أيضا تساعدنا، مو الأشياء اللي تساعدنا، اللي هي من المنتجات اللي نستفيد منها طبعا من الطحالب، اللي هي antimicrobials, antivirals, therapeutic proteins, drugs and antifungals and many more. هي لأنها جزء من المكونات الطبيعية، يعني ما هي كائن يعيش ويموت بسرعة كالحشرات أو الكائنات اللي تطير. تحتاج مثلا وقت طويل لبدء وإنهاء دورة الحياة حقتها، لذلك هي بشكل عام أنفع بكثير حتى من الإنسان نفسه. فنسبة إحداثها للمرض أو العدوى جدا قليلة، لكن تصنف من الكائنات المعدية. So they can cause less than any other microbe on our planet for infection. And they are good for the environment, good for the people, good as a source of oil and food, and also for cosmetics, pharmaceutical products, or industrial products, and also for إيش اسمه العلاج البديل أو العلاج الطب البديل، اللي هو يكون مثلا بمكونات من عشبة البحر، مكونات من عشبة داخل مثلا المحيط وإلى آخره، يعني حتى ما يحتاج أحط معاها مادة كيميائية، فهي علاجية أكثر من إنها معدية. ولكن but they are classified as microorganisms. They are not big enough to be classified as either plants or animals or humans. So they are microorganisms. Okay. Now I will explain what happened if you are confused about the bacteria or fungi or parasites or allergy or any microorganisms. They are in our planet and they are doing good for us, or they are doing the damage or make us lose our health. To explain this, I have to differentiate between microorganisms and normal flora. As I said before, some of the parts of bacteria, they are parts of the components of my body. Some bacteria are part of the component of shaping my liver or my lungs, my skin, my hair, my legs, etc. And these bacteria called normal flora, or we call it resident microflora. البكتيريا الطبيعية أو البكتيريا النافعة هي تكون جزء من تكوين العضو، جزء من تكوين الكبد، الأمعاء، الشعر، البشرة، الأظافر، القلب، الرئة إلى آخره. فلذلك هي نسميها بكتيريا نافعة لأنها موجودة فعلي ما تحدث العدوى. Normal microflora of the body is a normal bacteria lives and impacts the anatomy, physiology, and morbidity of the human body from birth to death. موجودة في جسمي من الولادة إلى الوفاة. ليه؟ لأنها جزء من تكوين العضو. We develop a varied microbial flora on our skin, and mucous membrane contains a large number of bacterial cells in and on our bodies, which helps formate the body shape, tissues, skin, and organs. The resident microflora of the body are the organisms that are on the body on a continual basis. ما أقدر أستغنى عنها وما أقدر أشيلها لأنها جزء من تكوين العضو. فوجودها ينفعني ويساعدني إني أعيش حياة أفضل. في بعض الحالات النورمال مايكروفلورا لما ما تكون موجودة أو غائبة تسبب اعتلال صحي. مثال، نولد كأشخاص طبيعيين عندنا بكتيريا في الأمعاء تهضم كل أنواع الأخضر واليابس اللي إحنا ناكله، ولكن في سن معين بعض الدراسات وضحت إنه لما يجتاز الإنسان عمر 25 سنة أو 26 سنة ويكون استهلك كل أنواع البكتيريا اللي في المعدة اللي تهضم سكر اللاكتوز، خلصت، استهلكها كلها، شرب حليب لين ماتت كل أنواع البكتيريا اللي في بطنه اللي تهضم اللاكتوز. لذلك لما يجي يشرب حليب يصير عنده حساسية ويصير الهضم أو عفواً عملية الإخراج صعبة بالنسبة له ويضرب عليه القولون فما يقدر يهضم الحليب. لذلك يحتاج يشرب حليب خالي من اللاكتوز، حليب خالي من سكر اللاكتوز، عشان معدته ما فيها بكتيريا تهضم اللاكتوز. فوجود البكتيريا اللي تهضم اللاكتوز يعتبر نوع من أنواع النورمال فلورا اللي موجودة في طبيعة أجسادنا تساعدنا إننا نعيش حياة أفضل. So what happened is the normal flora in our bodies works for our good. They are there because they are part of our body. As I was saying, the people after 25 or 26 age, the studies shows that some people has an allergy against the lactose, so they have no other bacteria in their abdominal area which can digest the lactose, so they have to drink free lactose milk or any food that doesn't contain lactose due to lack of bacteria that can digest the lactose that was in their body 25 years ago as a normal flora was created while they were born. So normal flora considered as a blessing for us. And as you are continuing living, maintaining your health, eating healthy food, maintaining movement or sports, or even also sleeping well, all of your normal flora will rest, will give you the good shape of the body, will give you the good skin that glowing, also will give you a good function inside. لذلك ينصح بالمحافظة على الأجزاء الطبيعية اللي موجودة أصلا في أجسامنا قدر المستطاع عشان نقدر نعيش برضو حياة أفضل، لأنها خلقت معانا وموجودة كجزء من أجزاء أعضائنا وموجودة للحفاظ على صحتنا بشكل عام. فأنا ما ينفع أروح أخربها وأخلصها مثلا، أو إني أخذ علاجات ومضادات وبوتوكس وتشميس ويعني أعطي نفسي يعني فرصة إني أدمر هذا الجسد اللي يعني جاء عندي كنعمة. فدائما النورمال فلورا يتم يعني خلطها مع البكتيريا كده بشكل عام، إنه كل أنواع البكتيريا لا خلاص هي ضارة وستحدث عدوى، لا، النورمال فلورا أو البكتيريا النافعة هي موجودة للنفع، جزء من مكونات العضو. بشكل عام النورمال فلورا تعمل علاقة جيدة مع جسم الإنسان، وبرضو تعتمد على منطقة وجودها داخل الإنسان، ولكن بشكل كمان عام آخر هناك مناطق في في جسم الإنسان ما فيها بكتيريا أبدا، لا نافعة ولا ضارة. مثال الدم ما فيه أي نوع من أنواع البكتيريا، والجهاز العصبي الأوسط، الجزء العلوي من الجهاز البولي، وتجاويف الجسم. هذه الأماكن نقية تماماً من أي نوع من أنواع البكتيريا سواء كانت نافعة أو معدية. فإذا جينا ناخذ مثلاً عينة من لعاب الإنسان ووجدنا فيها إيكولاي، هي إيكولاي ما هي جزء من مكونات المريء أو الحلق أو رأس المعدة وإلى آخره. فأنا حاعرف إنه هنا في خلل، مثلا غير موجود هذا الكائن بشكل طبيعي أو كجزء من مكونات هذه الأعضاء، عارف إنه هنا في خلل. طب أنا أصلاً قبل ما أخذ العينة لازم أعرف وأتأكد إنه أي عينة أحتاجها من جسم المريض كانت طلعت منه بسبب إنه في أعراض، في علامات signs and symptoms. لذلك قمت أنا أخذت عينة من المكان اللي يشتكي منه المريض، أو بشكل عام أخذت عينة دم، عينة بول، عينة مخاط، عينة لعاب، عينة براز وإلى آخره. صار عندي مريض في المستشفى عنده عرض، أخذت عينة، عرفت نوع الكائن، حعرف نوع العلاج بكل بساطة. So I was saying that the normal flora will make a good relationship with the host to make the host live longer and healthier life. And there are some parts of the human body that doesn't contain any type of bacteria. They are bloodstream, central nervous system, upper urinary tract, and body cavities. So if I had a patient with signs and symptoms, the doctor will ask for a sample, either urine sample, stool sample, blood sample. Then I did identify the microorganism or the bacteria in general. I will know if it's infection due to the signs and symptoms, or it's only a colonization. And also I will know this is part of the normal flora, this type of positive result from the lab. Is it normal flora or is it infectious flora or infectious bacteria? Or it's not normal flora in general. How I will differentiate between them, I will see the signs and symptoms. If they are only colonizing the bacteria in the wrong place, so it's not part of the organism, this is with signs and symptoms, so this is an infection. And we call it here transit microorganisms. They are present on a temporary basis. They may be encountered in our food, contaminated water, by handshaking, physical contact, or inhaling air particles that contain the infectious bacteria. Either way, I can find it in the environment, in radiology machines, in stethoscopes, in medical equipment, in the OR, in the ER, in the bed rails, in the surgical instruments, in the ventilators, etc. If I found the positive result from the lab as not a normal flora and the patient has a sign or symptoms, so this is called infection. For example, as I say, if I had a sample from the upper respiratory tract that contain E. coli, Candida species, Pseudomonas, Klebsiella, Enterobacteriaceae, and the people who are working in the lab will understand this. So this is not normal. This is not normal flora. This patient has infectious disease or infectious medical condition. As simple as it is. Also, in some samples, they may have fungi. As I said before, fungi is not part of the normal flora, except that they are part of the normal flora in the vagina. If I had a sample of liquid from the vagina and I found a candida, this is a normal flora, considered as a normal flora, and you can read about it more. So it doesn't only talking about the bacteria, either the fungi. يعني مو شرط كل العينات اللي تطلع منها كائنات معدية إنها تكون طبعا معدية، يعني ممكن تكون زي ما قلنا بكتيريا طبيعية. وأيضا في جزء من جسم الإنسان اللي هو المهبل عند السيدات، من الجزء المكونات الطبيعية فيه هو فطر الكانديدا، هذا جزء من المكونات الأساسية لسوائل المهبل. فأنا متى يعني أخاف على المريض أو أحرص على ظهور العينة في أسرع وقت ممكن، والمريض أيضا يكون حريص على نتيجة المختبر لما يكون في عنده أعراض. بمجرد ما تطلع الأعراض هو وقتها حيعرف إنه في حالة عرضية معدية أو عنده مرض معدي لا قدر الله. عشان زي ما قلنا نفرق بينهم بين الكولونايزيشن وبين الانفكشن. How to differentiate between colonization and infection. Now, I had a sample from a patient and he has an infection. He has signs and symptoms. And then I did found a large number of bacteria in my sample. And there is another patient that doesn't have signs and symptoms. And also I found the same or a higher number of clusters or of colonizing the bacteria in my sample. When I will call this an infection, when I will see the signs and symptoms. If there is no signs and symptoms, it's only colonization. And also for colonization, I have to prescribe the right medicine or the right antibiotic to make the bacteria die in this part of my body. كيف أفرق بين الاستعمار والعدوى؟ إذا أخذت عينة من المريض، أخذت عينتين من مريضين مختلفين، واحد عنده أعراض وطلع عنده في العينة تبع المختبر كائن معدي. مثلا خلينا نقول طلع عنده في عينة المختبر أخذنا من الاثنين عينة دم، والمريض الأول اللي عنده أعراض طلع عنده إيكولاي في الدم، فأنا كده حعرف إنه عنده عدوى. ليه؟ عشان أعراضه، عنده أعراض، عنده سخونة، عنده الله يكرمكم إسهال، عنده تعب عام، عنده صفار، عنده رشح، عنده إلى آخره. العينة الثانية من المريض الثاني برضو عينة دم وطلع عنده نفس الشيء، عنده إيكولاي في الدم. طبعا هي نادرا ما يكون إيكولاي في الدم إنه ما فيها أعراض، لكن لو حصلت عندي عدد كبير من الإيكولاي في عينة الدم للمرض الثاني، لكن المريض ترى طيب، حرارته طيبة، ما عنده ضغط، ما عنده أي مشاكل صحية في الأكل والنوم والإخراج، فهذا أسميه استعمار بكتيري للإيكولاي، لفصيلة الإيكولاي في دم هذا المريض. أحتاج بس أصرف العلاج المناسب لحالته، وقد يكون ما يحتاج أصرف علاج. في بعض الحالات حتخرج البكتيريا من لوحدها مع فضلات الجسم الله يكرمكم. فأفرق بينهم بوجود الأعراض، يعني نفس العينة من مريضين مختلفين وممكن يكون نفس نوع البكتيريا وعددها وكثرتها، لكن واحد عنده أعراض فذلك يعني عدوى، والثاني ما عنده أعراض فذلك يعني استعمار. كده بكل بساطة عشان تسهل عليكم أكثر. لا تناموا. in general أو بشكل عام، لما أعرف إني أخذ عينة وتطلع فيها يطلع فيها بكتيريا أو كائن معدي ليس من البكتيريا الطبيعية المفترض تواجدها في هذا المكان، هعرف إنه حيكون غالباً في عدوى. كما نشاهد في الصورة، جزء من النورمال فلورا في الجهاز العضوي في الجهاز التنفسي العلوي، ستافيلوكوكس سبيشيز، ستريبتوكوكس سبيشيز، هيموفيلس سبيشيز. جزء من تكوين البشرة ستافيلوكوكس سبيشيز. جزء من تكوين الجهاز الهضمي انتيروبكتيرياسي، ايكولاي، إيشيريشي. يعني عادي ايكولاي تكون في الأمعاء، في المستقيم، ممكن تكون لاكتوباسيلس، في عندي زي ما قلنا الكانديدا في المهبل، لكن مو عادي إنها تكون في البشرة، مو معقول أو مو عادي إنها تكون في الجهاز العلوي التنفسي أو في الجهاز الإخراجي وإلى آخره. تمام؟ إن شاء الله بس يكون واضح. So it's not normal to find some bacteria or fungi and sometimes some types of them in other places that are not considered as a normal flora. So here I can discover or can have a hint that this is infection. نخليكم تصحصحوا شوية. يلا اللي عنده سؤال. مين عنده سؤال؟ باقي لنا جزء بسيط في المحاضرة، بس يلا قبل ما نكمل مين عنده سؤال؟ Who have a question? We didn't finish. طب أنا أسأل، إيش رأيكم أنا أسأل؟ العدوى بأعراض الاستعمار بدون أعراض نعاملهم سوا؟ لا. They cause significant morbidity and mortality for those infected and increase the cost of care provided to the patient due to the extended hospitalization and the use of restricted drugs. يعني العدوى المصاحبة للمستشفيات خطرة جدا على المرضى، الموظفين، الزوار، العوائل، الأطفال والجميع بشكل عام، وممكن تنتقل كمان من المستشفى إلى الخارج فتنتقل إلى المجتمع، فهي تشكل خطر وتزيد من عدد الوفيات والمرضى في المنطقة اللي تكون فيها أعدادها كبيرة أو منتشرة بشكل كبير، وكمان تزيد من تكلفة الخدمات المقدمة كخدمات رعاية صحية للمريض. ليش؟ لأنه مدة تنويمه طالت، وكمان قد يكون في هذه المدة تم استخدام العلاجات أو وصف العلاجات غير مناسبة أو غير صحيحة له، فحالته ما تحسنت، بالعكس ساءت. وهنا برضو وقتها يقدر ممكن يكون عندي فيه أنواع بكتيريا أو فيروسات أو فطريات ذكية فتعمل لي مقاومة للمضاد المصروف للمريض، فيصير إيش؟ نوع العدوى مستمر وموجود في المستشفى، قد ينتقل لمريض ثاني، قد ينتقل لقسم ثاني. فإذا ما عملت الإجراءات الوقائية الكافية all hospital associated or hospital acquired infections will spread more, will have an outbreak, we will have a lot of problems. بعدين تقولوا إيش؟ infection control ما يسوي شغلهم، ما يعطونا educational lectures. يلا عندكم lecture الآن، حتى لو في المستشفى عندكم ما حد اداكم. hospital acquired infections or healthcare associated infections كيف تجي؟ as a direct result of the treatment or contact with health or social care setting. قد يكون جات من علاج خاطئ كده بسرعة، انصرف لي مضاد حيوي غير صحيح، فصار عندي مقاومة للمضادات وزاد عدد تكاثر البكتيريا داخل جسمه، أو كنتيجة سريعة أو يعني direct result of healthcare delivery in the community، يعني نتيجة مباشرة لرعاية صحية بس ما كانت داخل المستشفى، كانت خارج المستشفى وكانت خاطئة وانتقلت للمستشفى لأنه حالة المريض اتدهورت. so in the community، أو ممكن أصلاً تجي من الكوميونتي، تجي أصلاً من المجتمع. واحد كان أو واحدة عنده جاته قرصة أو اتقرص جاته ملاريا، أو واحد يعيش في مزرعة وجاته ليشمينيا عن طريق الفيكتورز زي ما قلنا، فتيجي من المجتمع إلى داخل المستشفى، ما ينتبهوا للأعراض، ما يستخدموا معدات الحماية الوقائية، المريض ما أخذ العلاج بالطريقة الصحيحة، فزاد عدد البكتيريا، زاد عدد الفيروس، انتقل للممرضة، الممرضة نقلته للمريض الثاني عشان ما غسلت يدها وما لبست القفازات، ما قفلوا الباب، ما عزلوا المريض، ما حطوه في نيجاتيف بريشر روم، يلا صار في عندي في المستشفى outbreak، أو صار عندي hospital acquired infections بعدد كبير. فهي كده تجي المستشفى بشكل عام بيئة معقمة ونظيفة، فقد يكون انتقال العدوى فيها بسبب ممارسات شخصية داخل المستشفى من الموظف وحتى من المريض، يعني مو بس نلوم الموظف. وكمان قد يكون أصلا هي جاية لمريض بسبب اللي هو ضعف مناعته، أعطينا علاج، عزلنا، عملنا كل اللي نقدر عليه، ضعف المناعة جاب لي كائنات opportunistic microorganisms انتهازية تستغل فرصة إنه مناعة الجسم مريض حقت المريض ضعيفة، يلا نستوطن الجسم ونعيش فيه ونسبب عدوى. فبشكل عام ما أقدر ألقي باللوم على أي أحد أو على أي سبب، لا، أنا أحاول ألم الموضوع وأعمل كنترول على الانفكشنز inside my hospital. كمان أحط خطط to stop spreading the infections. فإذا صارت عندي العدوى موجودة في المنشأة أو في مركز الرعاية الصحية، ما ألقي باللوم وما يعني أزيد المشكلة، لا، أول شيء أحل المشكلة اللي عندي وأعمل سيطرة على انتشار هذه العدوى، وكمان أحط خطة لإيقاف العدوى في المستقبل عن طريق إيش؟ عن طريق طبعا الإجراءات الوقائية، وحنتكلم برضو عنها في هذه اليومين إن شاء الله في المحتوى. من أشهر الكائنات أو العدوى اللي هي موجودة داخل المصاحبة للمستشفيات أو أيوة الكائنات بالتفصيل يعني أو بالتحديد اللي هي إيش؟ MRSA أو MSSA Methicillin Sensitive Staphylococcus aureus، هذه برضو مقاومة للمضادات. وعندي السي الكلوستريديوم تيفيسيلي، إيكولاي، سودوموناس، الكانديدا، النوروفيروس، الأسينوتوباكتر، وانتيروبكتيرياس. هي غالبا هذه اللي تكون داخل المستشفى، وغالبا تكون انتقلت عن طريق كونتاكت، عن طريق التلامس، المصافحة، أو مثلا لمس المريض بدون غسل اليدين، لمس أدوات المريض بغير تعقيمها، لمس جهاز الأشعة، إدخال القسطرة بدون تعقيم، وإلى آخره. فألاقي هذه الكائنات الدقيقة في أي عينة، عينة بول، عينة براز، الله يكرمكم، عينة لعاب، عينة دم، إلى آخره. So the most famous hospital-acquired infections, the microorganisms that caused hospital-acquired infections, and we know that they are in the hospital due to contact with others, either from the patient to the staff, or from the staff to the patient, from a machine to the patient, from the surfaces, from the door pins, or any other way. So they are transferring through contact transmission. They are Pseudomonas, E. coli, Acinetobacter, Candida. Candida no, ما هو بالcontact. Norovirus, C. diff, MRSA, etc. And we can find it in any sample, either urine, stool, sputum, blood, tracheal aspirate, either way, etc. So they are the most famous hospital-acquired infections. Some of the reasons causing hospital-acquired infections are invasive devices for patients in the ICUs. For example, indwelling urinary catheters, intravascular catheters, or mechanical ventilators. Any device will enter the body of the patient through intact skin or mucosal membrane. Why? Because there is something in the, or some element in the chain of infection called portal of entry. A door that is open in the human body for the microorganism to enter proudly and make the infection, do the infection, or do the damage. Also, other reasons or other devices like the nasogastric tube and the misuse of antibiotics, also the extended period of hospitalization days for the patient also will help in increasing the hospital-acquired infection. But the most main reason is invasive devices, because they enter the body of the patient. So من المسببات الأساسية والرئيسية لتواجد العدوى المصاحبة للمستشفيات، وخاصة في أقسام الرعاية الصحية الحرجة، اللي هي الأجهزة اللي تدخل جسم المريض، اللي تكون تحتاج إحداث فتحة للدخول إلى جسم المريض. هي المفروض تدخل عشان تحسن صحة المريض، ولكن إذا لم يتم تعقيم مثلا المكان اللي هو مكان الفتحة أو يد الممرض أو الممارس الصحي، ولما يكون الجهاز أصلا معقم، كل الظروف أو اللي هي كير باندلز يعني مطبقة بشكل صحيح، حيكون في عدوى، غالبا حيكون في عدوى. ليه؟ لأنه في بند من بنود سلسلة العدوى يسمى portal of entry، اللي هي فتحة لدخول الكائن المعدي، موجودة وجاهزة، اللي هي الديفايسز، اللي هي الأجهزة اللي تساعد المريض على تحسين صحته. غالبا حتكون القسطرة البولية، القسطرة القلبية، وجهاز التنفس. أي جهاز يدخل لجسم المريض عن طريق الغشاء المخاطي أو تلامس البشرة. كمان في اللي هو غذاء أو أنبوب التغذية اللي نحطه للمنومين من هذه الفئة، برضو يؤدي إلى نوع ما اصطحاب بعض أنواع الكائنات المعدية، وأيضا إساءة استخدام وصرف العلاجات بشكل عام والمضادات الحيوية بشكل خاص، وتنويم المريض لعدد أيام أطول من المعتاد، فهذا يدل على إنه حالته بتتدهور ما بتتحسن. وكمان يلعب دور سن المريض واستجابته المناعية لتحسين حالته أو تكوين كائنات يعني عفوا خلق كائنات حية دقيقة مقاومة للمضادات داخل جسمه أو لا. لكن السبب الرئيسي لتواجد العدوى المصاحب بالمستشفيات هو اللي هي الديفايسز، الأجهزة، اللي هي القسطرة القلبية والقسطرة البولية وجهاز التنفس. هذا آخر جزء في محاضرتنا اليوم، اللي هو شرح سلسلة العدوى. جهزوا أسئلتكم وإن شاء الله يعني ننتهي قبل الوقت بإذن الله. chain of infection هي عبارة عن 6 بنود لازم تكون متوفرة عشان أعرف إنه هذه ترى عدوى، بالإضافة إلى وجود طبعاً الأعراض اللي هي من ضمنها. فلازم يكون عندي كائن معدي. I have to find 6 elements to complete the chain of infection. Microorganism, which is virus, bacteria, fungi, parasite. And then I should have reservoir host or object number 1. The victim number 1. Animals, humans, food, water, either equipment, surfaces, even air, because some microorganisms can live in the air. And then I have to find a way out of the first victim. Why? Because he's already infected. So I have to go out from the body number 1 or victim number 1 to the victim 2. So I have to find a way to go out of the first host, which is I transfer through blood, mucus, or vomit, urine, feces, wound exuded, or anything, using method of spread, which is number 4. Either air particles, drops from the patient, liquids, body secretions, contact surfaces, or equipment. I will use this to move from body number 1 to body number 2, from patient 1 to 2. And to go inside the body, the number 2 or the next patient is a way into the body. So portal of entry and portal of exit. Number 5, portal of entry. I have to find a way to be inside the second body, which is can be broken skin or broken open wound, inhale contaminated air particles, ingest contaminated food and water, or through the mucous membrane in general. So number 5, I have to find a way inside the other body. Number 6, I have already infected the next victim, which is mainly will be in danger, elderly people, small children, infants, or family members in general. So I have to finish the 6 elements to complete the chain of infection. I have أحتاج أخلص 6 بنود من سلسلة العدوى عشان تكتمل عندي دائرة العدوى. غير كده ما يعتبر عندي سلسلة عدوى مكتملة، ولن تحدث العدوى ولن تطلع لي أعراض في هذا الموضوع. لازم يكون عندي كائن معدي اللي هو أول شيء بكتيريا، فيروس، فطريات أو طفيليات. تدخل للجسم الأول، وحنشرح فيها كيف دخلت للجسم الأول. الجسم الأول أو الكائن الأول قد يكون إنسان، حيوان، نبات، جهاز، سطح، أدوات، أكل، غذاء، ماء، إلى آخره. أي حاجة ممكن يعيش فيها هذا الكائن المعدي. بعدين لما يدخل الكائن الأول يحتاج يستقر في منطقة عشان يعرف يخرج من الجسم الأول، يغير البيئة اللي يعيش فيها. فلازم يتأكد من إنه في مكان في جسم الإنسان الأول أو الكائن الأول يقدر يعيش فيها ويرتاح ويتكاثر ويحدث الضرر، ويقول لك يلا من هنا أنا حأخرج. فهذه المنطقة اللي يخرج منها way out of the body أو portal of exit قد تكون إنه يستوطن في الدم، البول أو المخاط أو القيء الله يكرمكم، أو يكون في جرح مفتوح، جرح من عملية، أو جرح مفتوح بسبب خياطة سيئة. فأستخدم هذا المكان عن طريق الانتقال للجسم الثاني، إني أدخل فيه الجسم الثاني اللي هي ذرات الهواء، سوائل الجسم، دم بشكل عام، أو تلامس مباشر للسطح، للأيدي، للأدوات المستخدمة في العملية، مشيمة، إلى آخره. أي طريقة أنتقل فيها من الجسم الأول للجسم الثاني، وخاصة إني أنا لقيت مخرج في الجسم. هنا هذه النقطة اللي هي الدائرة الصفراء أو number 3 where the signs and symptoms will appear. فهنا أتأكد إنه هنا في عدوى. So way out of the body means that the virus has finished his job. He did take all of the energy of the antibodies and damage the immune system, and he will take all the power when he is going out, and his way out of the body meaning symptoms. هنا وقتها يخرج الأعراض سواء كان بكتيريا أو فيروس أو فطريات. لما تبدأ تخرج الأعراض معناته إن الكائن الدقيق المعدي بدأ يخرج من الجسم ويبقى يروح لجسم ثاني. فكيف يخرج؟ يسحب كل القوى اللي موجودة في جسم الإنسان، وأضعف نقطة في جسم الإنسان سواء خلايا ضعيفة، عضلات ضعيفة، قلة غذاء، فيتامينز، معادن إلى آخره، هي اللي يستوطن فيها ويبدأ يصير فيها الألم، وتبدأ تظهر الأعراض. يعني ليه؟ لأنها ما هي قادرة تقوم بدورها أو وظيفتها الإكلينيكية الصحيحة عشان يكون الجسم صحيح ومتعافي. فهنا تبدأ تظهر الأعراض مع ظهور الألم في هذه المنطقة، ويكون إيش الكائن المعدي طلع كل القوى اللي موجودة في الجسم مع وحارب الأجسام المضادة وأحدث الضرر قد ما يقدر عشان يطلع من الجسم الأول للجسم الثاني. في البند رقم 3 هنا حتبدأ تطلع الأعراض، ولما تطلع الأعراض معناته أنا عندي عدوى كده بشكل عام. واستخدم وقتها إما ذرات الهواء أو السوائل من الجسم أو التلامس المباشر. بعدين وأنا في طريقة انتقالي من الجسم الأول للثاني أدور على مكان أدخل منه للجسم الثاني. أي طريقة، سواء ذرات الهواء أو التلامس أو السوائل، أي شيء ألاقي منه فتحة تدخلني للجسم الثاني عشان أحدث الضرر والعدوى وأكمل الدائرة. حتى لو مو أنا، حتى لو السلالة حقتي اللي أنا قاعد أتكاثر بيها في الجسم الأول، مو شرط أنا. يعني الكائن المعدي قد يتوفى أو قد يموت في هذه المرحلة، لكن في معاه ما شاء الله الجماعة ما يقصروا. فأي فتحة أقدر أدخل فيها للجسم الثاني حأحدث العدوى. قد ما يكون العدوى في جسم، قد تكون في موية، في غذاء، قد تكون على سطح وإلى آخره. حتستمر العملية، هي مستمرة. portal of entry will be to the second victim، اللي هي فتحة لدخول في الجسم الثاني أو نقطة استقرار في المادة أو الجسم الثاني. قد تكون جرح مفتوح أو استنشاق الهواء اللي يكون فيه ذرات هذا الكائن المعدي، أو أكل غذاء أو هواء أو شرب ماء ملوث بكائنات أو جزيئات هذا الكائن المعدي وإلى آخره. مجرد ما أدخل إلى إلى ألاقي فتحة أدخل فيها الجسم الثاني، صار عندي اللي هي رقم 6. يلا الآن هذا الجسم أصابته العدوى، خلاص صار الجسم الثاني هو رقم 2 في إعادة سلسلة الدائرة. يلا نبدأ ندور على way out of the first one of the first body and then using a method to spread and then another body to be infected. أكثر الأشخاص يكونوا في الخطر إصابة بالعدوى اللي يكون عندهم جهاز مناعي ضعيف، كبار السن، الصغار، الرضع، أفراد العائلة بشكل عام، المدخنين، اللي تحت العلاج الكيميائي، اللي نقلوا أعضاء، اللي دائما يتبرعوا بالدم، يعني فئات مرة كثيرة. ولكن الوقاية يعني الوقاية وحماية الأشخاص عن طريق كسر سلسلة العدوى أو قطع سلسلة العدوى هو أفضل طريقة لأنه أنا أحميهم منها. الوقاية خير من العلاج دائما دائما دائما. في مصطلحات أو اللي هي تعريفات الانفكشن كنترول يمكن ما تكون واضحة، شكل الشريحة يعني ما هي مرتبة بالطريقة الصحيحة، أعتذر منكم. عندي المايكرو أورجانيزم نقول عليه infectious agent in infection control terminology. Reservoir host, it's the same. Way out of the first body, portal of exit. Mode of transmission, which is clarify the method of spread. Way into the second body, we call it portal of entry. And the second victim will be called as a susceptible host. It's a new host and the cycle will repeat again. Let's explain this in a simple way for more clarification. نشرحها بطريقة كمان أفضل وأحلى. عندي فيروس كورونا، إنفلونزا، H1N1، أي نوع فيروس. لما أصيب به الجسم الأول، أنا عندي الآن مايكرو أورجانيزم وعندي infectious agent وعندي reservoir host اللي هو الكائن الأول، الضحية الأولى. لسه ترى هنا لسه ما بدأت الأعراض، بس دخل للجسم الأول عن طريق إيش؟ portal of entry. لما نيجي ندور على way out of the first body, here the symptoms will appear. عشان يخرج يسحب كل القوة اللي معاه في الجسم ويطلع using method of spread, mode of transmission, which is here عندي drops, nose drops. Then I will find a new environment to live. So I have to find a new portal of entry to the second victim. لازم أدور على طريقة أدخل فيها إلى الجسم الجديد اللي هو مستخدم إيش؟ قطرات الجسم أو سوائل الجسم. فيجي المريض الأول يمسح أنفه بالسوائل اللي فيها، ويا عمري ما يغسل يده ويروح يسلم على الطرف الثاني، أو مثلا ينسى أو يستخدم قلم يكون فيه جزء من سوائل الأنف ويجي واحد وراه يستخدم هذا القلم وحوسه. المهم جاته هذه الجزيئات من الفيروس without protection, without hand washing, face mask, isolation or social distancing. So there is a portal of entry to the second victim. And then the second victim is already cooked. So now he has the virus. The chain of infection will be repeated again. I can break the chain of infection in each step, in each element. وأقدر أقطع سلسلة العدوى في كل خطوة. في كل خطوة أقدر أستخدم التطعيم، أستخدم العزل، أستخدم الكمامة، أستخدم hand washing، أستخدم القفازات، أعقم البيئة أو الغرفة أو العيادة، أعقم الأدوات المستخدمة. في كل خطوة أقدر أعمل breaking the chain of infection. هذه إن شاء الله حنشرحها غالباً بكرة بإذن الله. how to break the chain of infection أو في آخر يوم. For example, if I had a new virus, again for repeating the cycle or the chain of infection, I have a virus and he's already infecting the new or the first victim. And then the first victim was aware enough to wear a mask, only a mask. The chain of infection will be broken. So there is no chain of infection. قطع سلسلة العدوى ما يحتاج له ذكاء وعبقرية. إذا أنا كنت واعي كفاية أقدر أقطع سلسلة العدوى حتى لو مو أنا المريض. أنا دوري مثلا كممارس صحي أعلم المريض. فما يحتاج له ذكاء. هي العزل يكفي، التطعيم يساعد، غسل اليدين ممتاز، لبس أدوات أو معدات الحماية الشخصية excellent. Taking the medication is perfect, but in each point I can break the chain of infection. So if the virus had done damage for the first victim, and the first victim was aware enough by wearing the mask, he will stop spreading the infection. As simple as it is. In my hospital, as a healthcare practitioner or a healthcare worker in general, if I saw a patient with continuous coughing or sneezing, I can advise him simply, please break the chain of infection by wearing the mask. حتى لو كنت موظف إداري, even if I wasn't in the medical staff, if I was in the administrator, if I was a security, housekeeping, maintenance, anyone can advise the patient, please wear the mask so we can protect others by stopping the chain of infection or breaking the chain of infection. As simple as it is. يعني هي ما هي صعبة ولا هي يعني مرة تافهة يعني سهلة، ولكن هي بس يحتاج لها وعي. يعني أنا أعرف ترى لو حقطع سلسلة العدوى by doing this بس بكل سهولة وليس فيها أي نوع من أنواع الصعوبة. So as I was saying, I can break the chain of infection in each step. حقيقي. In the microorganism point or element, I can break the chain of infection. How? By the correct diagnosis and the correct treatment or medication. I can break the chain of infection by establishing the antimicrobial stewardship program in my hospital. So I can stop prescribing the wrong medication for the wrong patient and the wrong duration and the wrong time. And also I can manage the antibiotic resistant in my hospital. In the reservoir host, I can break the chain of infection by cleaning, disinfection, sterilization for the instruments, for the environment, for hand washing, everything. And also I can implement the infection prevention policies, and also can review the pest control. If I had a clear environment, I can ensure that this patient will not get infected. Also, in the portal of exit, the way of getting out element, I can break the chain of infection by hand hygiene, PPE, control of aerosols and splatters by wearing the mask, respiratory etiquette. I will explain it tomorrow by coughing or sneezing in your hand with washing or wearing a mask, or coughing or sneezing in your elbow, or isolate yourself. Also, I can break the chain of infection in this element by waste disposal. I can dispose all the elements or instruments that I was using for this patient who has infectious agent in the medical waste, not in the general waste. So I can cut or break any chance of continuing the infection. Also, in the mode of transmission, the way of the way of spreading the infection, I can break the chain of infection by hand hygiene. wearing PPE, food safety, cleaning, disinfection, sterilization, also isolation. I can isolate the patient who has the infectious agent. I can break the chain of infection in portal of entry or way of into the second patient by hand hygiene, PPE, personal hygiene, first aid, also removal of catheters and tubes that are not necessary. Why? Because it's a portal of entry to the second patient. So I can break the chain of infection here. In the hospitable host or in the second patient or second victim element, I can break the chain of infection by immunization. Immunization means vaccination, treatment of underlying disease, continue taking the treatment or the medication. Health insurance also will help the second patient in protecting himself against infectious diseases. And also patient awareness and education. So he will wear the mask, he will protect himself and his family by washing hands, for social distancing, wearing PPE, taking the vaccines, etc. In each step, I can break the chain of infection. في كل بند من بنود سلسلة العدوى أنا أقدر أوقف العدوى بالوعي، بالتعليم، بالممارسة، وحتى تبادل المعلومات. أسأل، أبحث. في بند الكائن المعدي أقدر أقطع سلسلة العدوى عن طريق التشخيص الصحيح وصرف العلاج الصحيح. لأنه لو جبت كائن طلع لي في نتيجة المختبر كائن معدي وبعدين رحت صرفت له العلاج الخاطئ، يا حيصير مقاومة للمضادات الحيوية ويعطيني سلالة جديدة من الكائن، ما حتتدهور حالة المريض فالعدوى حتزيد وقد تنتقل للمريض اللي جنبه وإلى آخره. أنا أقدر أقطع سلسلة العدوى بالتشخيص الصحيح وصرف العلاج الصحيح في هذا البند. وأيضا إنشاء برامج اللي هو الأنتي مايكروبيال ستوردشيب اللي هي تساعد في التقنين من صرف المضادات الحيوية للناس دون الضرورة أو الحاجة لها، أو للمرضى دون الحاجة لها. ما أصرف أي مضاد حيوي على طول كده بسرعة لأي إنسان ما عنده نتيجة مختبر تديني إنه في كائن معدي يحتاج فعلا هذا المضاد الحيوي. أيضا في بند الكائن المعدي أو الجسم المعدي الأول، أقدر أقطع سلسلة العدوى عن طريق تنظيف وتعقيم البيئة والأدوات، عن طريق إني أتبع إرشادات وتعليمات مكافحة العدوى، وإني أنا أسيطر على موضوع البيست كنترول. البيست كنترول هذا حد إيش؟ الحشرات، تنظيف البيئة اللي أقدم فيها الرعاية الصحية، والتخلص من هذه الكائنات اللي قد ممكن تنتقل إلى المريض وهي أصلاً عندها كائن أو عندها مثلاً فرصة لنقل العدوى. في بند اللي هو نقطة لخروج الكائن الحي، كائن الدقيق الحي من الجسم الأول إلى الجسم الثاني، أقدر أقطع سلسلة العدوى عن طريق برامج نظافة اليدين، برامج معدات الوقاية الشخصية، عن طريق respiratory etiquette اللي هي تعليم المريض آداب العطاس، إنه يعطس في يده مع غسل اليدين، أو يلبس كمامة، أو يعطس في منديل، أو يعطس في الكوع. وطبعا هي الأفضلها إنه هو على طول يغسل يده في جميع هذه الخطوات. إني أعلم المريض هذه الآداب اللي هي اسمها آداب العطاس. وأيضا أقطع سلسلة العدوى في هذا البند عن طريق waste disposal اللي هو أي شيء يخص هذا المريض اللي عنده عدوى، الأدوات استخدمها، معدات إلى آخره، أتخلص منها في فين؟ الكيس اللي فيه medical waste، نفايات طبية. ليه؟ لأنه فعلا هذا الكائن عنده، هذا الإنسان عنده عدوى، فبلاش أعدي فيها موظفين الهاوس كيبينج ولا المرضى التاليين أو أي أحد من الستاف يجي يلمس أي شيء بالغلط. وكمان حتى طريقة التخلص من هذه النفايات تكون بطريقة طبية مدروسة. فالويست ديسبوزل أو ويست سيجريجيشن يهمني إني أنا أتخلص من النفايات المستخدمة والأدوات والمعدات المستخدمة على المريض اللي عنده كائن معدي في النفايات الطبية. لأنه هذا يساعد في قطع سلسلة العدوى. أقدر أيضاً أقطع سلسلة العدوى في بند mode of transmission أو طريقة الانتقال عن طريق إعادة برامج نظافة اليدين، لبس معدات الحماية الوقائية، انتبه لسلامة الغذاء، إنه يكون عندي نظيف جدا لأنه هو طريقة أو وسيلة سلامة الغذاء وشراب الماء برضه، لأنها وسيلة لانتقال العدوى للمرضى داخل المستشفى، وأيضا التعقيم والتنظيف والتطهير وعزل المريض المصاب بعدوى. وهذه فعلا حتكون يعني أساسية في قطع سلسلة العدوى في هذا البند. في بند وجود فتحة لدخول الكائن المعدي إلى جسم المريض، أقدر أقطع سلسلة العدوى برضو عن طريق نظافة اليدين أو برامج نظافة اليدين ولبس معدات الحماية الوقائية والنظافة الشخصية للموظف وللمريض، للممارس الصحي وللمريض. أو يكون عندي مثلا first aid اللي هي حقيبة الإسعافات الأولية، إني ألبسه كمامة بسرعة، أعقم له يده بسرعة، أعقم له المنطقة اللي مثلا أعطيله فيها إبرة السكر، زي كذا portal of entry. هذه اللي هي تفرق، تقدر تقطع سلسلة العدوى عن طريق. أو كمان نقطة مرة مهمة اللي هي الاستبعاد أو إزالة أي نوع من أنواع الأجهزة والأدوات، الأجهزة اللي تدخل جسم المريض اللي هي القصرة البولية والقلبية وجهاز التنفس، أو اللي هو أنبوب الغذاء. إذا ما يحتاج المريض على طول ريموفيت على طول. ليش؟ لأنه يساعد في قطع سلسلة العدوى. آخر نقطة اللي هي break the chain of infection in the susceptible host اللي هو الضحية الثانية اللي حتروح لها العدوى. هنا كيف أقدر أقطع سلسلة العدوى؟ عن طريق التطعيم، عن طريق علاج المرض المعدي ومو أترك العلاج، وأساعد المريض إنه يكون واعي كفاية لأهمية أخذ العلاج، وعن طريق التأمين الصحي أو التأمين الطبي اللي هو يساعد المريض مادياً ويدعمه نفسياً في إنه حيتلقى العلاج والرعاية الصحية الطيبة، فيقوم إيش؟ يحتمي في هذه الحالة ويتأكد إنه إن شاء الله بإذن الله حيتم شفائه، مو يكون في تحت ضغط مادي ونفسي وجسدي وإنهاك يعرضه للمزيد من العدوى. كمان الهيلث انشورنس أو التأمين الصحي يساعد المريض إنه هو ياخذ علاجات أفضل. وكمان عن طريق تعليم المريض وعائلة المريض اللي هو تقديم المعلومات الكافية لأنه هو يعرف ترى لازم تقي نفسك من العدوى عن طريق التطعيم، واستكمال جرعات التطعيم والعزل ولبس الكمامة وغسل وإلى آخره. الدور الأساسي في عملية التعليم له يعني حيصنع فرق مرة يعني كبير. زي ما قلنا في سلسلة العدوى ممكن في كل بند أقدر أقطع سلسلة العدوى بمجرد إني أنا عرفت وتعلمت إني أنا أقدر أقطع سلسلة العدوى في هذه المنطقة. إن شاء الله يكون الشرح شافي وكافي ووافي. يلا if you have any questions. الله يعافيكم جميعا. يلا يلا نجاوب دون الأسئلة عشان نجاوب ونطلع. If you have any questions, we will answer your questions so we can finish our lectures. If you have a question, write it all down. What do you mean by ranking a PPE? ما فهمت. Ranking of PPE, please. هذه ما فهمتها صراحة. ما أقدر أطلع questions. طيب حطوا الأسئلة هنا في الشات، إيش رأيكم؟ You can put questions in the chat, it will be easier. هل ممكن نصرف مضاد حيوي إذا لقينا التهاب بالأشعة فقط بدون شكوى أي أعراض أخرى؟ أكيد لا، لأنه لازم تحليل. لا، يفضل لا، وطبعا يرجع لي حالة المريض والطبيب، بس يفضل لا. الآن الأنتي ميكروبال ستورشيب صارت كلها مقننة بشكل مضاعف عشان ما نضر المريض. ترى ما نصرف المضاد الحيوي مو عشان إحنا ما عندنا مستودع فيه مضادات حيوية أو إحنا ما نبغى نكلف على المريض أو التأمين يدفع، لا، عشان سلامة المريض. لأنه الآن صاروا يعني عالميا توقفوا عن إنتاج المزيد من أنواع مثلا المضادات الحيوية، خلاص اكتفوا باللي موجود، لأنه كل ما نسوي مضاد حيوي جديد تزيد مقاومة المضادات باي المايكرو اورجانيزمز. الأجيال الجديدة من الكائنات الدقيقة ترى ذكية، ترى زي أجيالنا اللي موجودة يعني كبشر. كل ما جاءت أجيال جديدة كل ما خربت على القديم وصارت ذكية. كنت بقول مثل يعني كل ما جاء أجيال جديدة حتسوي إيش؟ يعني تطلع أذكى من القديمة. كل ما جات سلالات جديدة زي فيروس الإنفلونزا، كل ما جاني سلالة جديدة خربت الجهاز المناعي بطريقة جديدة واحترافية وذكية أكثر من الأجيال السابقة. فكل مضاد حيوي أنا حيطلع لي مقاومة من الأجيال الجديدة، فتوقفوا عن إنتاج المضادات. لذلك إحنا ما نصرف أو تم تقنين صرف المضادات الحيوية. فما ينصرف مضاد حيوي بدون تحليل، على الأقل لازم تحليل وأشعة أو تصوير ألتراساوند أو إلى آخره. ولكن كده بدون أي سبب لا، ما يصير. ولو أنا كنت المديرة يعني ما حمشيها. إذا كان عند الشخص مرض مناعي وحصل له استعمار نعامله بنفس الطريقة ولا يعامل معاملة خاصة؟ لا، يعامل معاملة خاصة. مرام was asking if the patient has an immuno condition or a special immunocondition disease and he has a colonization, we should treat him as the same as the other patients? No, he has a special condition. We have to find out all of the samples. We need urine samples, sputum, blood, all samples عشان نحمي المرض قد ما نقدر. بعدين نقرر إيش العلاج اللي نصرف له. Can we get the certificates هناك عند الدعم الفني الشهادات؟ هل نوع المرض المعدي بيأثر على التخلص من النفايات؟ نوع المرض المعدي يأثر من أي ناحية؟ هي النفايات فكرتها إنه we have to segregate waste. if I did finish all the يعني procedure of medical and general waste segregation, all of the process will be correctly. So نوع المرض المعدي يعني يأثر على الخطورة حقت إني أنا كيف أتخلص منه، عشان في نفايات طبية تنحرق وفي نفايات طبية تروح مع النفايات يعني بشكل عام في طريقة تخلص كيميائية أخرى. فمو نوع المرض المعدي، هو نوع المرض المعدي يختلف، يؤثر على الخطورة إنه كيف ينتقل عشان أعرف أتخلص منه يعني بطريقة لائقة بيئيا فما أضر الناس. نعتبر high infectious disease like measles، نعتبر كل النفايات خطرة. هل تختلف عن مريض الإنفلونزا؟ لا، كلها نفايات ميديكال. شوفي أنا بالنسبة لي الأبيض أبيض والأسود أسود. ميديكال ويست يعني ميديكال ويست، يعني infectious waste. لا تلعبي فيها، لا تقولي عشان هذا عنده مرض معدي أخف ضرراً لا 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 لا. ميديكال ويست أي شيء يلمس المريض، سائل، كونتاكت أو من الغشاء المخاطي، سوائل أو دم أو أي شيء يخرج من المريض medical waste. I consider it as a medical waste. ليش؟ عشان ما نشكك نفسنا وندخل في فتاوي ما لها داعي. Anything discharged from the patient, touch the patient, considered for me as a medical waste. هل تعتقدين كثرة التعقيم والوقاية ممكن يكون سبب في زيادة مقاومة البكتيريا؟ لا، مستحيل، عمره ما يكون. نترك مجال أن الجسم يتعرض للعدوى؟ لا 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 لا. هذه حكاية مين يسأل؟ اسمه سامي. هذه حكاية إني أنا أختبر معدتي، أقوم أكل مجرثم ولا أكل من الحجة عشان أختبر أمعائي ومعدتي. لا لا، هذه الحركة لا لا، ما هي جيدة. بالعكس، التعقيم والوقاية عمرهم ما يضروا. وهي didn't give children under 18 superfluxin. هذه تسألي فيها الدكاترة والصيدلية، ما هو تخصصي يا روان. أنا ما أعرف إذا أنا أعطي سوبرفلكسن لأقل من 18 أو لا. نرجع للشات، إذا عندكم أسئلة اكتبوها في الشات، أوكي؟ أو شاركونا مواقف أو أسئلة يعني، أو مواد مثلا حصلت لكم نستفيد منها. لا ما انتهينا لسه. we have